Hello, good morning, everybody. Can you hear me?
Yes, good morning.
Good morning. So
Good
today
morning.
is
Good afternoon.
World Wetland Day. That is Feb 2nd. As usual, we are meeting today after 23. In 24, we are meeting for the first time. And I am happy to see that we have something like 40 participants. They have already joined us from many countries. Today we have a symposium, virtual symposium, which is having participants from Nepal, India, Sri Lanka, Philippines, Taiwan, and Thailand. And hopefully Micronesia, I'm not sure. 6 countries we have already confirmed. And we have more than 25 presentations approximately. If we take all these 25 presentations, we need to have at least 3 to 4 hours. So what we will be doing, we will make a break for 5 minutes for after 1 hour, for 30 minutes or so and so, so that 5 minutes we can get relaxed and then again we can start. We have received the name of the participants for which we are preparing the certificates. And if there is any additional names to be added or any change in the spelling of the participants, please send us today afternoon, because this is the morning time in Delhi. Okay, you can send us the changed names so that we can send their certificates well in times, so that the students are happy. We are going to have this regional meeting, as you know, April 15 to 18, and hopefully that will be in Malé, in Maldives, which is a very beautiful, wonderful country. And I'm not sure if our host, they are here. I will request Rifat or somebody, Balsma or somebody from his office who can really uh, give us some brief on that also. But one thing I would like to mention, we have received only few names from few countries regarding the participants. Please send us the name of the country representative who would like to attend the regional meeting, because that is very important for us, so that we can get the approval of GIO for the purchase of the air ticket and also to make the booking of the hotel accommodation in Maldives. But anyway, regarding Maldives meeting, I will speak to you after we finish this presentation, which is quite a, a long day we are going to have. And I have host here, Hina and Sonal. I've asked them to be the co-host and the host because I will be around, but little bit busy with some other issues which I have very recently. And then I hand over to the host, Hina and Sonal, to start with the presentation, and uh, let us go to the sequence which we have already circulated to you. And also I will request all the participants uh, to follow the time, around 8 to 10 minutes together, including questions. As you can see, we have large number of presentations which will take more than 4 hours today. And I can see that we have something like 56 participants, 55 participants who have joined us today. This wonderful number in the countries we will see. So I welcome all of you to the new year with greetings and then happiness and the strong globe activities in 2024. So I hand over to my hosts, Hina and Sonal. Please. Okay.
Thank you, sir. Good morning, everybody, and welcome to the Asia Pacific Virtual Wetland Symposium. As you already know, we have circulated the list of schools that we are going through during the symposium. So first, we'll start with Sri Lanka, Truston College, and the presentation title is Beniwal Athriti Sanctuary by Ayodhya, Devinder, and Aloka. Are you guys ready with the presentation, or do you want me to share it? Because I have your presentation with me.
And what I suggest, if you don't have presentation, if you inform me in advance so that your presentation is ready.
Yes. Okay, good.
And also one more, we request you to put the name of the country in front of your name, like Sri Lanka people they have done, so that we know how many countries we are here today. Please put up the name of your country in front of your name. You can start.
Yeah, you can start Sri Lanka.
Our presentation, our research about the wetlands in Sri Lanka for the Wetland Day 2024. Our main line is wetlands are for human well-being. Moving on to the next slide.
Sorry to disturb you, Elisa, but you are speaking very slowly. We can't hear you properly. If you could speak closer to the mic or.
Okay, I'm sorry. Yes, we are from Sri Lanka, Thurston College, Colombo. We are here to present our presentation that we made for the wetlands of Sri Lanka for the Wetland Day 2024. And in the first slide, you can see our team, my fellow teammates, Ayodhya Dwara, Aloka Pansilu, and I am Asala Girihagama under the guidance of our teacher in charge, Mrs. Supeksha Pesega. In the first slide of this presentation, we are going to talk about what are wetlands. The Ramsar Convention, which was formed in 1971, has defined what are wetlands. This definition as follows: the areas of marsh, fen, peatland, or water, whether natural, artificial, permanent, or temporary, with the water that is static, flowing, fresh, brackish, or salt, including areas of marine water, the depth of which at low tide doesn't exceed 6 meters. And moving on to the next slide. Speaking about the major wetlands in Sri Lanka, Sri Lanka has some significantly beautiful wetlands. Bodhana National Park is one of the major wetlands in Sri Lanka and also a huge attraction by the tourists and locals. The Anavilundao Lake Sanctuary, which was a huge topic in Sri Lanka recent days, and the Madure Wetland, Vankalai Sanctuary, Kodumbigala Wetland Group, Vilapathu Wetland Group, and today our special topic, Bellangwila Athiria Sanctuary, can be mentioned as the most important wetland located in Colombo district. And now we are moving on to our main topic today, Bellangwila Athiria Sanctuary. My fellow teammate, Aloka Pansilu, is now ready to give you guys a proper detailed introduction about the Bellangwila Athiria Sanctuary. Aloka?
Yeah, thank you, Asala, and good morning, everyone. Here we are talking about Bellangwila Athiria Sanctuary, which located in Western Province and Colombo district as well. 0 meters to 6 meters altitude. It consists of 372 hectares. Let me show you the landscape of the sanctuary. Here we can see some urban areas in Colombo district, such as Borlasgamua, Artidia, Bellangwilla, Verahara, and Divulapitiya around the sanctuary. Fortunately, the sanctuaries still consist with their biodiversity among those high populated areas with some facts. If we move on other side, we can see a thematic map. According to the map, we can see some cultural features such as population density, agriculture land use pattern, and infrastructure facilities. Now we can see Bellangwilla Athidiya Sanctuary wetland, which based on Ramsar classification. As an overview, primarily a freshwater marsh ecosystem surrounded by a rapidly developing urban landscape, as we mentioned. The marshes serves as an important flood reduction area, which provides protection to the surrounding inhabited areas. An important stopover for a human number of migratory birds, an important roosting site for herons and egrets, identified as an IBA by Bird Life International. SLR.
Yes, as we looking at the physical features of this Bellangwilla Athidiya Sanctuary, this Bellangwilla Athidiya Sanctuary is interconnected to the floodplains of the Bolgoda system through the Bolgoda Lake, Bolgoda Canal. Bolgoda Canal is also a huge part of Sri Lankan ecosystem, and also several other man-made canals connect to the Athidiya marshes, to the Colombo marshes. Bellangwilla Rajma Vihara, which is a very special religious place for Sri Lankan Buddhists, is also located close to this wetland. Is another special feature. A small man-made lake is also located within the wetland system. It is a special fact. And also must mention that this Athidiya Sanctuary, Bellangwilla Athidiya Sanctuary, is located within the wet zones and receives a rainfall of 2,500 to 5,000 millimeters per year. And it holds an average annual temperature around 27 Celsius and also an average humidity around 80%. Moving on to the next slide. Here we are going to take a look at the ecological features of this sanctuary. Our teammate Ayodhya is now ready to give you guys some detailed introduction about these ecological features of the Bellangwilla Athidiya Sanctuary.
Thank you, Vasil. And we had look on the physical features, and now we are moving on the ecological features. Consists of shallows, freshwater ponds, marshes, and seasonally flooded grasslands with scattered shrubs and small trees. Significant biodiversity, including 77 species of butterflies and 37 species of dragonflies. 5 species of butterflies are considered nationally threatened. 15 species of nationally threatened and endemic vertebrates have been recorded from the wetland. Bird life is over 168 species have been recorded. More than 30 species of freshwater fish have been recorded, including several exotic species. As we spoke about natural beauty of this area, we can't forget about hundreds of thousands of endemic species which house in this area. First, you can see endemic fish species near from this slide. Next one also is special the huge part of ecosystem amphibians. You will be able to see some endemic include like the amphibians, Bufo atucorale, Polypides cruciger, and more. Next, aquatic reptiles there are including freshwater turtles, serpents, and crocodiles. As we mentioned, this place is a house for a lot of immigrant bird species and endemic bird species, and also we can't forget about water birds. Water birds include uncommon species such as Pelicanus philippensis, Rallus striatus, and more. And also we can see some rare endemic mammals as we mentioned this slide. Asala?
Yes, Ayodhya. from some detailed ecological features about this Belangul Athiri Sanctuary. Now we are moving on to talk about the disturbance and threats. As a student who live around this sanctuary area, I've seen these disturbance and threats with my own eyes. Factories in the vicinity release effluent waterways passing through the marsh, which then enter the Bolgoro Lake. As I mentioned, Bolgoro Lake is also a huge part of Sri Lankan ecosystem, so it also damage those factory effluents are damaged to the ecosystem more and more. And the Dehiwala Mount Lavinia Municipal Council, these cities are very populated urban cities, uses the northern and eastern parts of the marsh for garbage dumping. Such factors have an impact on the waterbirds' habitat quality and cause large-scale fish kills. And also road kills on the main roads. Consumption of the meat from terrapins and birds cause decline in these species. In addition, ecolocation is a threat to the bird populations inhabited in marsh, and also habitats have been lost due to the clearing of shrubs, construction purposes in the surrounding areas, as well as human encroachment into the marshes from its periphery. Ongoing landfill marshlands, largely with household activities. And here are some pictures that we were able to capture with our camera, which show you guys how this sanctuary area has been affected with the bad human activities. Moving on to the next slide. As our last title, we are going to talk about the significance of wetlands in Sri Lanka. Sri Lankan wetlands have some significance to them. All wetlands comprise of a combination of soils, water, plants, and animals. The interplay between these elements allow wetlands to perform several functions that are beneficial to humankind while generating healthy wildlife forest resources. Fishing industry has spread widely around the Belangul Athiri wetland, and people also do industries like flower trading. Flower trading is a very famous industry around this wetland area because of the Belangula Rajamahawihara temple. The combination of these functions together with the rich biological diversity and cultural heritage of wetland makes this ecosystem invaluable to people all over the world, and the loss of wetlands results in the loss of these benefits, leading to harmful consequences on the human welfare and to the extinction of species that are ecologically dependent on the wetlands. Here in this slide, you can see some pictures that we were able to capture, people doing their industries which are depend on the sanctuary area. So as the end, all we have to say is we should protect this sanctuary because it is a huge attract by the tourists and the travelers all over the world and Sri Lanka. It's not much interested by the locals, but it is an icon of Sri Lankan identity and also very helpful to Sri Lankan human industries. We shouldn't protect this just because it is an identity or help for our life. We must protect this because it is a huge part of our ecological ecosystem. It houses for so many migrant birds, migrant animals, and so many endemic animals. So that's why we must protect this area, not just for humans, for our ecosystem. Thank you.
Thank you, Sri Lanka. Thank you for the wonderful presentation. Is anybody has any question to the participant regarding their presentation?
Let them write in the chat box. No?
Yeah, but we don't have any question in the chat box. That's why I'm asking if anybody has any question. If not, you can keep your question in the chat box and we will get back to them after the next presentation. Thank you. Thank you.
Wonderful presentation with the beautiful photographs of the study site. Congratulations, Sri Lanka team. I think this is the first time we have these Sri Lanka presentations in our symposium. Thank you very much. And maybe we can proceed with the second presentation.
Yes.
Thank you, Dr. Desh.
Hi.
Yes, I am just going to remove all of you from here. Okay. So the next presentation is also from Sri Lanka in Mahinda Valley Gale School. or college. I'm not sure whether it's not written in there. And the participants are E.K. Tawashi, WPGM Mauri, and I.K. Donga. Sorry if I pronounce your name wrong. And the topic is Madhu Ganga Ramsar Wetland. So are you guys ready, or should I share the presentation for you? Okay, wonderful.
Okay. Wetland Day 2024 Wetlands and Human Wellbeing. We are from Mahinda College, Goa, Sri Lanka. Teacher in charge, Mrs. Nadika Sajivani. I'm Tavish Dinushan. Murubashita, Danuja Sadeva, and Netum Divsara are members in my team. First, let's see what a wetland is. Wetlands are areas that are inundated throughout the year or periodically inundated in the natural environment. Estuaries, lagoons, deltas, coastal marshes, inland reservoirs, etc. belong to these wetlands. Next, let's see what types of wetlands there are in Sri Lanka. The first is freshwater wetlands. Inland reservoirs, rivers, lakes, outcrops, freshwater marshes, etc. belong to this. Next, brackish water wetlands. These include brackish water wetlands with salinity intermediate between brackish water and freshwater. Deltas, estuaries, mangroves, lagoons can be given as example. Marine wetlands. Oceanic ecosystems like coral reefs, salt marshes, seagrass beds, etc. belong to this. Brackish marshes is next type. This type of wetlands can be seen in Mutturajavila area. Finally, artificial wetlands. These wetlands are created by human intervention. Paddy fields, aquatic ponds, lakes can be mentioned as examples. Wetlands in Sri Lanka. Next, let's examine the wetlands in Sri Lanka. As far as Sri Lanka is concerned, wetlands are of very high value. To date, 36,900 wetlands have been identified. Among them, 6 are Ramsar wetlands. They are Wankale Sanctuary, Wilpattu Ramsar Wetland, Anavilundawa Tank Sanctuary, Madhuganga, Boondala and Kumana Wetland Cluster. Next, I invite Manula to continue the next part.
Thank you, my dear Tavish Dinushan. Next, we will talk about the Madhuga wetland, that is a Ramsar wetland. First, about the location. It is located in southern province in Goa district, and its latitude is 6 degrees 17 minutes 50 seconds north, and the longitude is 80 degrees 3 minutes 16 seconds east. This wetland is located in Balapiti area, about 47 kilometers from Goa city. Madhuga wetland was officially declared in 2003 by Ramsar Convention as a Ramsar wetland. There are 64 islands here, but today it is reported that some of these islands have been submerged due to rising water levels. Among them, the smallest island is called Satapaha Island and the biggest island is called the Madhuga Island. Here are some of the unique places and things we can see in this wetland. Kodduwa Temple, Mahakapnawala Sima Malagaya, and Ratamila trees. In the first picture, we can see the Kodduwa Temple, that is a religious place of Buddhists. And in the next picture, we can see an shrimp enclosure. And in the third picture, we can see the Satapa Island, that is the smallest island of this wetland. And in the last picture, we can see Ratamila trees, that is not inherent to Sri Lanka, but in Sri Lanka we can see only in this tree, only in this wetland. Biodiversity of Madhuga wetland. Wetlands are very rich in biodiversity because they provide food and habitat for many species. Some species not live in wetlands, but they provide their food from wetlands. This wetland is home to 303 plant species and 248 vertebrate species. Although there is no air in the soil, plants with different adaptations are in this wetland. Fishes, oysters, various insects, shrimps, amphibians, and reptiles live in this wetland. This wetland is also give the foods, habitat, and breeding grounds for migratory birds. Next time, right, my dear friends, Dhanitya Sadev to continue the presentation.
Thank you, Manula. First of all, let's see some native plants that can be seen in this Madhuganga Ramsar wetland. In the first picture, we can see Bovidia. As we all know, Bovidia is the provincial flower of the southern province in Sri Lanka. Then the second picture shows Kakuna. This plant belongs to endemic according to its habitat. In the last picture, we can see Kurundu. Kurundu is mainly used for flavoring of foods. Not only that, it is a very good exporting item in Sri Lanka. Then let's move to a new topic. That is about some native animals that can be seen in this wetland. As the first picture shows, we can see Kaluandura. It is an native animal that can be seen in this wetland. As well as it is also known as purple-faced langur, and it belongs to the order primates. Next one indicates Dandulena. It is also known as Ratufa macrora as the scientific name. Moreover, it is the national animal of Sri Lanka. Then the last picture shows loris. We can see this animal only in the night time as it is a nocturnal animal. Then let's identify some benefits of these wetlands. First benefit is obtaining water for daily needs. As water is our main necessary, we obtain water from many ways. This is also a one major way that we obtain water. Then the second benefit is taking different types of medicines. A lot of herbs for local medicines can be found in this area. Some of them are oatmeal and vatakeya. Then the third benefit is use the clay collected in exhausted for industries. People make bricks, tiles, and pots from those clay. Then the next benefit is to carry out some activities like fishing industry and agricultural industry. Then the next benefit is obtaining raw materials for local industries. The last benefit is for tourism. Next, I invite Netum to continue the presentation.
Now we will look about current status of Madhuganga wetland. At today, wetlands are under various threats due to adverse human activities. Some of them are reclamation of swamps, dumping of garbage, industrial waste, and chemicals into wetlands. Pollution from agricultural runoff, industrial discharges, and urban activities can introduce harmful chemicals and nutrients into the water, negatively impacting water quality. This can have detrimental effects on the health of the river and its ecosystem. Damages to aquatic life due to illegal boating. Increased tourism can lead to habitat disturbance, pollution, and other negative impacts. If not managed sustainably, boating activities in particular can disturb wildlife and contribute to erosion. Damage to environment beauty due to informal constructions of shrimp cages, deforestation of wetlands, and destruction of mangroves, and also dumping of sand. After that, we will look about another threat, that is invasive plants. Here are pictures of some invasive plants. The introduction of non-native species can disrupt the natural balance of the ecosystem. Invasive plants or animals may outcompete native species, leading to biodiversity loss. Then let's talk about how to protect wetlands. Proper use of wetlands can protect them for future generations. What can be done for that? Carrying out development works according to formal plans. Nowadays, we can see the nature is polluting because of informal plans. So, development works must do according to formal plans. And also, strategies like not dumping waste into water sources, reducing the use of chemical fertilizers, and also conserving wetlands can be used. Local and national authorities, environmental organizations, and communities need to work together to implement strategies that ensure the long-term health and sustainability of the Malu River ecosystem. Thank you.
Okay, this is wonderful presentation of the Madhu Ganga wetlands. This looks very well done, very well preserved wetlands, and I think we should plan some activity in Sri Lanka maybe next year or later this year so that we can have the opportunity to visit some of these wonderful wetlands. Thank you very much, the students from Sri Lanka, for this wonderful presentation and also the coordination which you have among yourself. Thank you. Can we have the next presentation?
So the next presentation is also from Sri Lanka, from Sujata Vidyalaya in Kirikala Kele Wetlands.
A very good day to all of you. My name is Nohimsa Vijayatunga and I'm from Sujata Vidyalaya Matara, Sri Lanka. First, let me introduce you to my teammates. Mithuli Dulhara, Lihini Dahamya and Saheli Okitma. And we all are here to present to you about Kerala Kale Wetland. So, I'm going to begin my presentation by explaining to you what a wetland is. So a wetland is an area where the water covers the soil or it is present either at or near the surface of the soil all year or for varying periods during the year, which includes the growing season. And these wetlands may support both aquatic and terrestrial species. So estuaries, lagoons, deltas, coastal marshes, and inland reservoirs are some examples of wetlands.
I think Sri Lanka se.
So there are many wetlands in Sri Lanka, and they can be categorized into three main parts, which are inland freshwater wetlands, saltwater wetlands, and man-made wetlands. First, we have inland freshwater wetlands. So these wetlands consist of rivers, streams, floodplains, freshwater swamp forests, villas, peatlands, and freshwater marshes. So these wetlands provide many benefits, which includes water supply, biodiversity conservation, and cultural value. Then we have saltwater wetlands. So these include seagrass beds, coral reefs, mangroves, salt marshes, lagoons, and estuaries. So these wetlands, they have this unique flora and fauna adapted to high salinity, temperature, and sunlight. And finally, we have man-made wetlands. So man-made wetlands in Sri Lanka consist of hydropower reservoirs, irrigation tanks, reservoirs, and rice fields. So did you know that Sri Lanka is believed to have the highest number of man-made water bodies globally, estimated to be over 10,000? So let's get into our topic, Kerala Kale Wetland. As you can see in this slide, it is located in southern province in Matara district, and it's 3 kilometers away from Matara town. And Kerala Kale is a wetland covered with mangrove forest in the south of Sri Lanka and stands today as the key habitat for wetland biodiversity in the country. And it lies between one of the longest river in the country, known as Nilwala River. Now, my friend Methuli will explain to you about the biodiversity in Kerala Kale. Over to you, Methuli.
Thank you, Nahimsa. As she mentioned earlier, my part is to make you guys aware about the biodiversity of Kerala Kale wetland. So, it can be categorized into two parts, which includes fauna and flora. And let's move on to our first part, which is the diversity of flora. 13 species of mammals, 103 bird species, 48 wetland bird species, and 25 freshwater species have been recorded from this site. Many species of butterflies, reptiles, amphibians, and hundreds of invertebrate species also decorate the biological wealth of Kerala Kale. So let's glance at these pictures. At first, we have purple face leaf monkey, lesser winda, then we got purple heron, chocolate macaque, and finally you can see the snakehead. So let's move to the diversity of flora in Kirla, which consists of 83 plant species belonging to the 40 family. As you can see in this slide, Kirala, Olu, and Karankapu are some of the popular food items in there, while popular Ranking Kiridia are also popular of some of the medicinal plants which are also popular in there. So my part is over from here.
อ๋อต้องเป็นเทคโนคณะในนี้แน่เลย
Saheli, can you take your part? Can you take your chance and make us aware about your part, please?
Sure, Mithili. Kiralkala is one of the prominent tourist destinations in Mathura district, Sri Lanka. This ecotourism area has much to offer to tourists with its wetland ecosystem consisting of housing birds, butterflies, animals, beautiful plants as well as rare plants. A boat trip through the canals of Kiralkala is an experience of a lifetime too. Not only that, Kiralkala has been a source of resources for the people who are living around for decades. People use Kiralkala for several reasons, including commercial, agricultural, recreational, educational, and entertainment purposes. Commercial purposes include fishing and harvesting of lotus stems and flowers. Agricultural purposes are paddy cultivation, coconut, banana, and vegetable cultivation. Recreational activities such as picnicking, jogging, and exercising are also popular. Enjoyment includes photography and birdwatching. And finally, Kiralkala provides a wonderful research and an educational center for students all over the country. Over to you, Lini.
Thank you, Saheli. So I'm going to make you all aware about the threats that have affected Kiralkala wetland. The application of pesticides and weedicides to crops and overgrazing can cause destroyal of fauna and flora there. Porcupine, wild hare, white bow, grey-headed swamp hen, and a few duck species are hunted for human consumption. Excessive freshwater fishing has also decreased fish density within the sanctuary. The road system expansion within the sanctuary blocked the flying routes of birds and regular routes of other animals. Many peacocks, pigeons, doves, mayas, and waterhen were harmed due to collisions with highway vehicles. Backcourt digging and removal of mud and aquatic plants allowed intrusion of seawater into marshland. So the death of hundreds of fish over months is common in the sanctuary. But don't forget, I have suggestions on protecting Kiralkala sanctuary also. First of all, we have to make sure that the current state of solid waste management is maintained thoroughly. A very aggressive invasive plant, Velata, can be found along the irrigation canals. Taking necessary management actions could stop this from being a serious threat to Kiralkala. So taking actions against those who break the rules implemented to ensure the safety of flora and fauna in the area is suggested. Now we have come to the end of our presentation. Proper management of Kiralkala is important for conserving the biological, hydrological, and ecological aspects and for the surrounding communities to continue to use it in their daily lives. It is our responsibility to ensure that our flourishing and valuable wetland, Kiralkala, is well taken care of. Thank you.
Okay, this is again another wonderful presentation from Sri Lanka. And you can see how beautiful Sri Lanka is. This is wonderful island. And I must congratulate all the students for this. This is the third presentation. All the three presentations have been very well done. Congratulations to you for all these wonderful presentations. I think we have one more from Sri Lanka or two more?
Two more
presentations.
Yeah.
Two more. Okay. Hi.
And wonderful for the last team because they have still got 2 minutes left of their time slot and they finished early. So congratulations on that also. So the next college is Richmond College and they have a presentation on Buena Vista Coral Reef Wetlands. So Richmond College, you can share your presentation now.
I can see Asha in the photograph. Okay, start please.
Wetlands Day 2024, Wetlands and Human Wellbeing. This presentation is presented by Nithul Sathiragar, Sunala Sandhiyuru, Chandula Sara, and Senuddin Nadan, with the guidance of the teacher in charge, Mrs. Ranjani Karyavasam, from Richmond College, Galle, Sri Lanka. Good afternoon, everyone. I'm here to present a simple presentation on the wetlands of Sri Lanka, especially about the Buona Vista Coral Reef in Rumasala. This presentation will describe about the biodiversity, economic value, threat space, waste protect, etc. So, without waiting any further, let's get started. Let's first take a look at what a wetland is. Simply, wetlands are areas of land that are either covered with water or saturated with water, either permanently or seasonally. They have a rich biodiversity and high economic value. There are three main types of wetlands, as inland freshwater wetlands, marine and saltwater wetlands, and man-made wetlands. Let's take a look at each of them separately. First of all, let's focus our attention on inland freshwater wetlands. These are wetlands with water in them or on them most of the time. They are not connected to the ocean. Next, it's about marine and saltwater wetlands, which are wetlands with salty water from the sea or the ocean. They include areas such as lagoons, estuaries, mangroves, seagrass beds, and coral reefs. Finally, we have man-made wetlands. These are areas of land that have been artificially covered with water to suit human needs. These are primarily reservoirs. Now we can get started on the Buona Vista Coral Reef and its significance as a natural wonder. Chand will continue regarding the importance and biodiversity from here.
Thank you, Nitu. Buona Vista Coral Reef is a diminutive but exceptionally biodiverse ecosystem situated at the base of the Rumasala mountain in Ghost district, Sri Lanka. It encompasses an area of approximately 1 square kilometer within the 8-meter barometric contour and harbors a plethora of fish and invertebrate taxa, including some that are new to science. The reef is exposed to various anthropogenic and natural stresses that threaten its ecological integrity and aesthetic value, despite its significance for conservation and ecotourism. The reef remains unprotected and vulnerable for further degradation. The Buona Vista Reef exhibits a remarkable degree of biodiversity and endemism within its diminutive spacious extent and hosts a plethora of fish and invertebrate taxa, including some that are new to science. It also has a high diversity of coral genre species, with at least 2 corals that are undescribed and endemic to this reef. The reef is a hotspot for biodiversity and potential source of naval genetic and biochemical sources. However, it is also subjected to various anthropogenic and natural stresses that jeopardize its conservation and ecological resilience. The Buona Vista Coral Reef is a vital marine ecosystem that supports the livelihood of local communities that depend on fishing and tourism. The reef is located at the base of the Rumasala mountain, a scenic and historic site that is associated with many legends and myths. The reef is a potential attraction for ecotourism as it offers a unique opportunity to observe the rich and diverse marine life in a relatively accessible shallow location. The reef is a precious natural heritage that needs to be protected and conserved for the benefit of the present and future generations. Now I will invite Chanu to warn us about the threats faced by Buona Vista Coral Reef.
Thank you, Chandu. Even though the Bona Vista coral reef is a wonder of the natural world, it is facing many threats that threaten to destroy it. They can be classified as pollution, human encroachment, and climatic changes. This coral reef is a natural wonder, but it is under a serious threat from pollution. Pollution from land-based sources such as industrial waste, sewage, and agricultural runoff, as well as from marine activities such as fishing, boating, and tourism, have degraded the water quality and harmed the reef. Pollution affects the reef in many ways, such as causing diseases, algae growth, coral death, and reducing biodiversity. These effects reduce the economic and ecological value of the reef. We need to act now to stop the pollution and save the reef. The reef is also under constant threat from human encroachment. The human encroachment refers to expansion of human activities and settlements into natural areas such as forests, wetlands, and reefs. Human encroachment affects the reef in many ways, such as destroying the habitat, reducing the space, and increasing the disturbance. These impacts lead to loss of coral cover, diversity, and economical value, and make the reef vulnerable to each other threats. We need to act now to protect the reef from human encroachment. Climate change is a global threat that affects not only humans, also plants and animals. One of the most vulnerable areas is the coral reef, which provides food, protection, and habitat for many fish and invertebrates. This reef is under serious threat from rising sea temperatures, coral bleaching, and ocean acidification. These factors have caused the reef to lose its color, structure, and functions and put its survival at risk. We cannot afford to lose this natural wonder. Sunil will now tell you all about protecting this reef.
Thank you, Senul. Many efforts have been made to protect this reef, such as assessing the reef and recommending measures to prevent mining, water pollution, and overfishing by the Global Coral Reef Alliance. Studying the status of biodiversity and raising awareness by the Nature Conservation Department. Proposing to establish a nature park and a marine sanctuary by Department of Wildlife Conservation. We need to do all we can do to protect this reef. Now, let's take a look at what we can do as citizens of Sri Lanka to help in protecting this reef. We can avoid coral mining, dynamite fishing, and other harmful practices that damage the reef and its inhabitants. Reduce water pollution by disposing of waste properly and using eco-friendly products that do not contain harmful chemicals. Support conservation efforts by joining local groups, volunteering, or donating to organizations that work to restore and protect the reef. Promote awareness and education by learning more about the reef and its importance and sharing this knowledge with others. Practice sustainable ecotourism by visiting the reef responsibly, following the guidelines and respecting the wildlife. Thank you all for dedicating your time and listening to this presentation.
Hello, wonderful. Thank you for making wonderful presentation. All the presentation from Sri Lanka, they are wonderful. I must congratulate all of you. I will put up my comments in the end, and also thank you again for your teachers and your country coordinator and all the those who has guided you to make this wonderful presentation. Thank you.
Thank you, Dr. Desh.
Yes. You want to say something? Go ahead, please. Okay. If you don't have, then we go to the next presentation. Also, that is from Sri Lanka.
Yeah, that is from Sri Lanka.
The last one from Sri Lanka.
Last one from Sri Lanka, yes. And yes. Okay, so from Victoria from New Zealand, she has a comment about the presentations, that all the presentations are excellent. So, and they indeed are excellent. So congratulations to the team who have presented yet. And the next presentation is from DS Senakaya College on Digam canals.
Yeah, I think you can see my screen. Yeah. So guys, good morning to everyone. So I think you all know about Sri Lanka, right? So you know about our fascinating waterfalls, different types of wetlands here, marine wetlands, estuaries, lacustrines, and also we have Ramsar wetlands. We have 6 of them. I think you all know about them, like Boondara National Park, Anavilundao Sanctuary, Kumara National Park. So you guys all know about those things, right? So, but we are just not today here to present you something like that. That means that everyone know. So yeah, what's about today's presentation? Yeah, it's about, as you can see in the screen, Diagama Ala Wetland. Yeah, so I know everyone in the meeting doesn't know about that, even Sri Lankan. So that's the most interesting part here is going on. So yeah, let's continue. We are the Science Society of DSNR College, Colombo 7, Sri Lanka, with Mrs. Danusha Kumari, teacher in charge, and I Mudul Mitra Abesinghe with Shashit Lakshita Angunavala and Tharukaransa Taberatna. So I would like to invite Tharukaransa Taberatna to continue this. Tharukar?
Okay, thank you, Udule. So, here this slide represents us some photographs of a wetland. It's a rich ecosystem full of rivers and we can see water refilling as well as water running. Moreover, the flowery, rocky and muddy pools, as well as floodplains. Especially, we can see some kind of mangrove systems which stabilize the river banks. Okay, so next we are going to explore where Diagama Ala is. So, all you know about our motherland Sri Lanka, right? As well as the Colombo, which is the commercial capital of Sri Lanka. Well, so the Diagama Ala wetland is located in Diagama Gramaniladari division, which owns to Homagama Divisional Secretariat Administrative Unit. Furthermore, for your reference, we provided here Google Map tags to find this wetland easily. You might think this wetland is as big as Kumana, Alavilundava, and Poondala, but it is not that large. The expanded area of this wetland is just 2 square kilometers. Okay, then we could explore the biodiversity with my mate Udula. Udula?
Yeah, thank you, Tharukar. So the most interesting part, everyone's, biodiversity. So let's talk about that. Yeah, first we have fish, the etiology in the area. So first of all, this area is actually highly diverse in freshwater fish diversity. So all the details here you are seeing in the screen is from etiological research, which is actually done by the Environmental Society of DSNR Ecology 2022. All right. So yeah, according to that result, here we have total of 21 native freshwater species in this wetland, and 3 of them, as you can see in the right side of the screen, are actually endemic and globally and nationally threatened species. The first one is Latka Varuna. He's a critically endangered. That means CR classified species according to the global red list, and the second one, Hordadia atukorali, which is a VU categorized vulnerable species according to the global red list. And the third one, Devarium micronema, which is also called as Kitulgala danio, which is an endangered species, also categorized as in EN category. So yeah, this is the thing actually we are trying to explain you under these 8 minutes. the valueness of this ecosystem. You can't find those species around the world. Even in Sri Lanka, it's really hard to find those things, those species. But you can see them in Diegamala wetland. But anyone actually don't know about those things. So that's why we are trying to explore those things to the world. So yeah, let's move into the next slide. The birds. Yeah, when it's come to ornithology, in this area, actually, you can see some two species of kingfishers and some species of egrets. But the real thing is, there is no any ornithological research has been done in this area before. So we can't say what are the exact species that who are just inhabited in this area, because there's a really need to do something about like that. Researchers, there's a great potential. All right, I'll mention it in the end of the presentation. So yeah, next move into the plants. So I would like to invite my fellow Shashit Lakshita to continue this. Shashit.
Thank you for the introduction. So whenever we are talking about plants under the category of biodiversity, we are in sort of information because there's no research done under the topic of plants. So it was a huge problem to identify whether they are native or endemic or the status of them, how valuable they are. So we have a huge room to do research about these plants in this wetland area near future. So in Society DSNI College have already conducted a research about fish in this area or aquatic animals, and we have mentioned some common plants we can see in this area. In this presentation, you can see like Ceylon swamp lily, hairy fig, yellow bamboo, green bamboo, Singhaladil, and Godamidella. And as Udula mentioned earlier, some notable plants we can see in this area like mangroves, water lilies, and various wetland grasses. And move on to the next slide about threats we have identified in this area. You can see we have done this research in 38 sites. Out of 38 sites, means most of the site is done. Have the threat of improper usage of agrochemicals. So that's the huge problem we can see in here. So, this area is situated in Homagama, which is quite popular for agriculture in Colombo district. So there are actually massive number of cultivation areas. So the agrochemical usage is high. So the lack of education to the farmers cause some huge problem to this area. And the next threat we have like scavenger, African jellyfish. So they are actually impacting the native biodiversity and causing unbalance of ecology and the disruption and loss of habitat to the native animals. And public awareness programs to educate people about how sustainable this environment is, how sensitive, how valuable they are. So that's it, and I'd like to invite Udula to conclude this presentation.
Yeah, thank you, Shashi. This is the end of the presentation, but we have to say something to you lastly. That is, as we were saying, mentioned for you, that is, this is not something that everyone in the world, even in Sri Lankans, know. Many of people don't know about those things, but it's highly valuable for our environment. So I think you also understand that one. So I think here, guys, you have. I think, guys, you are just interested in doing some researches about ecology, hydrology, zoology, anything. So this is a great place to conduct your research, to do your studies, because this is an untouched and just unrevealed place hide in the world. So it is our responsibility to explore these to the world. So yeah, that's all about it. And I would like to invite you all to visit this environment, Diegamala Wetland, and join with us. Thank you very much.
Hello. Yeah, I think it's wonderful. We had 5 presentations from Sri Lanka. So we
Dr. Desh.
say that how good these presentations are, the photographs, and then the coordination among the students, and then it was wonderful, and I enjoyed it. Thank you very much, Sri Lanka. Thank you, Sri Lanka Globe Coordinator, and
Thank you, Dr. Desh. Thank
presentation.
you, Dr. Desh, for your nice comments. Our country coordinator, Dirukshi Madam, has a family emergency today, so she is unable to participate. Thank you very much for giving this great opportunity to Sri Lankan children. Thank you very much, Dr. Desh.
And I think this is the result of the workshop which we had in Hanoi as the capacity building project. Am I right?
Yes, yes, Dr. Desh.
So that is the success of our capacity building project that we have 5 presentations from Sri Lanka. At this stage, I would like to mention that in Hanoi, we will be having students exchange activities sometimes, which I will explain at the end of this event. Thank you very much, Sri Lanka. Can we have
Thank you very much, Mr.
And thank you, Dr. Desh, and all, and especially for our students and teachers for these wonderful presentations. Thank you.
Thank you.
Do we have next country?
So next we have Taiwan, and again I also like to say thank you for the presentation, Sri Lanka, and we could see the passion in the students, and they are all passionate about their topics that they have chosen. And yes, so the next is New Taipei Municipal Defang High School, and their presentation topic is the impact of Pilomelo peel on Aedes albopictus larvae.
No, we can have the group photographs.
Oh yes, yes, you remind me correctly. Okay, so Sri Lanka, can I take 1 minute from you if you could stop your share, and we would have a group photograph, and after that we will resume with the presentation. Sorry for disturbance, just a 1 minute thing. It will be just 1 minute thing.
Open your cameras, please.
Yeah.
Open your cameras. उसमें आ जाना.
I would like the request to students that they will open their camera so that we know who are all presented and everything. Thank you.
Asha, you have not opened your camera. Asha, open your camera.
Yeah, everybody, we request you to.
Please, everybody open the camera. I don't see. Okay, Rod, how are you?
Okay, just give me 1 minute.
Take 2, 3 couple of these pictures.
Yeah, I have to because there are 4 pages. Okay, again. Okay.
Okay.
Yeah, thank you everyone. We have done. Thank you.
Okay, now we start with the Taiwan presentation.
Yeah. So Taiwan, you can share your presentation again. Thank you.
Hello everyone, my name is Bo Chun Lai and they are my classmates Yunsheng Jian and Zixin Zhou. All of us are from Taiwan and we study at Danfeng High School. Today we will introduce our research, the impact of Pilomelo peel on Aedes albopictus larva.
Mid-Autumn Festival is a traditional Chinese holiday characterized by family reunions and moon appreciation. It is also a season of abundant pomelos, which are believed to have mosquito-repelling properties. We are curious about the actual efficacy of pomelo in repelling mosquitoes.
This is our experimental diagram. First, we identify the species of mosquitoes. Later, we measure the pH and dissolve after of the.
Little louder, please.
We want to know which part of the pomelo pill is mosquito bearing. So we measure the breath-holding time, survival rate, heartbeat frequency, and wiggling frequency in outer or inner pomelo pill juice. Here is our research method. First, we go to grandma's garden, scoop water from the water barrel every week from November to December in 2023. Then we measure the pH and dissolve adjunct of the water sample. We identify the mosquito species and count the number of larvae. We use larvae of 0.4 to 0.6 centimeters for the following experiments. We prepare 10 grams of pomelo peel, outer and inner peel, with 400 of water in a juicer, resulting in a solution with concentration of 9%. Dilute to different concentration of pomelo peel juice and add them to test tube containing mosquito larvae.
We measure the breath-holding time, survival rate, and heart rate and wiggling rate of a mosquito larva. Breath-holding time calculate the duration it takes for larva to submerge, remain without breathing, and resurface. B. Survival rate. Observe the percentage of survivors after the experiment. C. Heart rate. Measure the number of heartbeats per second under microscope. D. Wiggling rate. Count the number of times the tail touched the head within 10 seconds.
Here's our results. We found 116 AED abapitus larvae using the Gold Arbusel app. The AED abapitus larva, also known as the Asian tiger mosquito, is a vector mosquito species. The adult mosquito has a distinctly white stripe in the center of its thorax. Male mosquitoes have feathery antennae as shown in the photo, while female have tail-like antennae. The larvae of AED abapitus have short respiratory tubes, and when they are come to the water surface to breathe, their bodies are oriented vertically with respect to the water surface. The pH is 7 to 6, 7.6 to 8.9, and dissolved oxygen is 7 to 9 milligrams per liter in the habitats of AED abapitus larvae.
Here is our result of the pomelo peel juice and the breath-holding time of AED abapitus larva. From the chart, it can be observed that as concentration of pomelo outer peel increase, the breath-holding time becomes longer. Conversely, when we use pomelo inner peel juice, the breath-holding time becomes shorter. These two results are different.
Here is our results of the pomelo peel juice in survival rates of AED abapitus larvae. From the chart, it is evident that as the concentration of pomelo outer peel juice increases, the survival rate decreases and all individuals die the next day. On the other hand, the inner peel juice has no effects on AED abapitus larvae.
Here is our result of pomelo peel juice and wiggling frequency of the AED abapitus larva. From the chart, it can be observed regardless of whether it is outer peel or inner peel juice. The higher concentration, the more wiggling occurrences. Furthermore, the wiggling frequency increases even after 30 minutes.
Here is a result of the pomelo pill juice and hard base of AED of Peter's larva. From the chart, this can be dissolved less than 9% concentration of other pill juice increases hard base frequency. However, when comparing the effects of 9% and 1.1% concentrations of inner pill juice infusion with the control group, it is found that there is no significance of impact of hard base frequency of AED of Peter's larva.
From the experiment, we have learned that first, AED albert can be found in the vegetable gardens of New Taipei City, Taiwan during November to December. Second, Aedes albus live in habitat with neutral pH and moderately high dissolved oxygen levels. Third, familial outer peel have more impact on the Aedes albus larva and inner peel. Fourth, familial outer peel can increase the breath holding time, heart rate, and wiggling frequency, but decrease the survival rate. Thank you for listening.
Thank you for such a wonderful and scientific presentation. And also it's interesting to know that how the juice outer peeling and the inner peeling behave in the mosquito. That is very scientific activity. I must congratulate three of you for such a wonderful and scientific presentation. And I am sure that you are going to be great scientists, the group scientists in future. Thank you very much. Maybe we have the next from Taiwan.
Yeah, we have another 5 from Taiwan, and thank you, New Taipei Define High School. Interesting presentation and interesting topic as well to use pomelo peel and see how the mosquitoes breathing and everything changes on the presentation. And the next presentation from Taiwan is National Liu Dong Senior High School, and their presentation topic is the quality of agriculture irrigation water in Taiwan.
Good afternoon. We are students from Taiwan Lodong Senior High School, and I am Han Chen-yun.
I am Lin Xiangjun.
I am Na Yousen. Our project focuses on the quality of agricultural irrigation water in Taiwan, specifically in Beichenzhen Canal in Lodong Town. The picture shows us where Taiwan is. Taiwan is a country surrounded by water. So we want to know the quality of irrigation water that will go into the ocean. And first, we look at the right side. We conducted the experiment in the middle reaches of Beichenzhen Canal on weekdays for about 3 months. From September 21st, 2023 to January 21st, 2024. And we turn back to the left side. We measured water depth and collected samples from the canal. Then we conducted tests to get some data. After getting the data, we could analyze it.
Now, let's talk about our results. The picture shows water temperature. X-axis means date, and the y-axis means temperature degrees Celsius. As we can see, the water temperature has decreased due to winter. ranging from 17 to 30 degrees Celsius, with an average of 23.73 degrees Celsius. The picture shows pH values. X-axis means state and the Y-axis means pH values. According to the picture, the values are divided into 3 parts because we use 3 different kinds of devices. We found that the value 208 measured was gradually on the increase. We thought it was strange, so we replaced it with 208. However, the measured values had too large a range, so we switched it to 206, and the values stabilized. The average is 7.43 except 208. TDS stands for total dissolved solids. The picture shows the values of conductivity and the TDS. X-axis means state and the Y-axis means values of conductivity and the TDS. Seeing from the picture, we notice that there is a relevance between conductivity and the TDS. When conductivity values drop, TDS values drop too.
And now, this is our another project. Water impurities observing and filtering. The picture on the left side is the tool used to filter the impurity in the water. And the picture on the right is one of our members using the microscope to watch the particle field and to calculate how many particles in the field paper. And every week, we filled 200 millimeters of water and calculate the sum of particles longer than 2.5 micrometers, which are visible under the microscope. The graph shows the sum of particles of every week. The value on October 25th was the lowest, and the value on December 27th was the highest. We noticed that there are obvious differences between the values, but we don't know why yet. We will keep observing and will understand it in the future. And this picture shows some of the impurities built on the field paper under the microscope on December 27, whose value was the highest. In conclusion, we compare the value we measure with Imagination Water Quality Centers in Taiwan. We found that all of the value meets the standard. Our report ends here. Thank you for everyone listening.
Thank you for wonderful presentation on the water quality of the river and the canal. As you know that in the countries the water is very polluted, but like in Taiwan, good. And congratulations for a wonderful presentation, the Taiwan group. I know you do a lot of scientific activities and also to the country coordinator and the Tina for helping us to get these presentations from Taiwan. Thank you very much. How many we have? 8 or 9 presentations from? How many we have presentations from Taiwan?
We have total 6 presentations from Taiwan.
How many?
6.
Okay, Now we have the third one now.
Yeah. The third one is from Koishang Municipal Jizen Senior High School, and their topic is exploring factor affecting the status of upside down jellyfish in environmental changes.
Hello everyone, we are from Shenzhen High School in Kaohsiung, Taiwan. Today we are going to report exporting factor affecting the status of upside down jellyfish in environmental changes. Here are the contents: research motivation, research purposes, research methodology, research findings, and conclusions. The first is our motivation. Upside down jellyfish grows in Kaohsiung Lingyuan Ocean Wetland Park. We are curious, why do jellyfish appear there, and what factors contribute to their living? So we divide it into 4 parts. First, to observe upside down jellyfish tissues. Secondly, to investigate the water quality in Lingyuan Ocean Wetland Park and upside down jellyfish. Next, to study the effect of light on the contraction behavior of upside down jellyfish. Lastly, to analyze the effect of nutrient deprivation on upside down jellyfish. On the following slide, jellyfish will be used to stand for upside down jellyfish.
Next is our research methodology. First, how to observe jellyfish tissues. We use a microscope to observe jellyfish tissues and mucus to find zooxanthellae and cnidocytes. Secondly, we want to know what kind of water factors influence jellyfish survival. So we're studying the water quality at 4 check spots in Lingyuan Ocean Wetland Park. Thirdly, we know from the literature that jellyfish umbrella contraction is related to preying activity. Then, we let the jellyfish exposed under 6 light modules to record the contraction times. This is our experimental procedure, and we repeat it 10 times from step 1 to 6. Then, we want to know what kind of time the jellyfish will be most active. We record the contraction times in the morning, noon, afternoon, and night. Lastly, from literature, we can know that if the jellyfish cannot get the food by itself and cannot get a nutrient by zooxanthellae either, it will go through autophagy to survive. So, we record the jellyfish umbrella size for 6 days.
We found that environmental changes can lead to nasal-side emission eject. The percentage comes down from 33.9% to 11%, with a difference by 22%. The dark yellow and the brown ones are zooxanthellae. The white ones are empty nasal-sites. The green ones are intact nasal-sites. This is a 10 minutes before and after picture. When we put the fish together with the jellyfish, we found that the jellyfish will secrete mucus to get out the fish and then poison them. When we put the mucus under the microscope, we found that there were not only nasal-sites but also contact zooxanthellae and the other organisms. There is the investigation in water quality. Among the 4 spots, A, B, C, D, spot C has the most jellyfish, and the water temperature and pH value of spot C are found to be lower. From the right chart, we can see that jellyfish are fit for growing in slightly alkaline water.
Thirdly, from the figure on the left, we can see that the strong magenta light triggers the jellyfish the most, and smaller jellyfish contraction times is more than bigger one. From the figure on the right, we can see that the jellyfish contraction is most frequent at afternoon. Lastly, after analyzing the effect of nutrient deprivation, From the table below, we find that if the jellyfish stay without light and food after 6 days, it will shrink more than without food but with light. From the experiment, we know that if the jellyfish not only cannot get food by itself, but also cannot get the nutrient from photosynthesis, it will shrink about 12 to 17%, more than the jellyfish only without food, which shrink about 10 to 13%. And then we come to the conclusion. 1. Water evaporation and cnidocytes. Water evaporation causes jellyfish to eject cnidocytes. With fewer water around, cnidocyte is activated. 2. Mucus, nutrients, and organisms. We find that there are many organisms in the mucus, and it's because the mucus contains many nutrients. 3. Temperature and pH values. Jellyfish live better in weak alkaline zone and the water temperature from 90 to 26 degrees Celsius. 4. Magenta light and contraction. Regardless of size of the jellyfish, strong magenta light proved to be the most effective in stimulating umbrella contraction. Besides, the highest contraction frequency occurs between 4:30 p.m. and 6:30 p.m. 5. Food, light, and autophagy. When the jellyfish live without light and food, it goes through autophagy and shrink to be a larger degree than that without food only.
And since the accidental discovery which the 95 exact under the microscope, we can continue to design experiments to explore this in the future. 2. The nutrient content in the mucus also needs to be further studied. 3. After interviewing experts, it is suggested that creating a beneficial environment to raise jellyfish would contribute to community tourism. Besides, developing ecological education and environmental guidance is indispensable. This is the end of our presentation. Thank you for listening.
Okay. This is wonderful. Jellyfish. I remember jellyfish is very, interesting for the kids, and the pond is very interesting. We saw jellyfish pond in Pulau, and this is very interesting presentation. Thank you very much. How you guys make such a scientific presentation? I'm very surprised, and also the teacher who guide you is great. The next one?
Yeah, the next one is also from the same school, and their topic is biomimicry, the combination of euphemism, alcoholism, and peanut shells.
Okay, please.
Oh, combination of
Wait. Ladies and gentlemen, good afternoon. We are students from Tianzhen Senior High School in Kaohsiung City, Kaohsiung, Taiwan. The topic we want to report is biomimicry, the combination of acornia crescents and peanut shells. Let's talk about our motivation first. In order to achieve a balance between scientific and technological industrial development, and the environment, we decided to explore into the problem of water pollution. To see the current status of water pollution in Taiwan, we take data of Gaoping River for research, because parts of the water in Kaohsiung City comes from this river. This picture is the data collected from the 5 monitoring stations along Gaoping River. it can be seen that Gulfin River is severely polluted. Here is an actual case of serious water pollution in Taiwan. Here we would like to introduce our biological inspiration Econia crescens. Its group temperature is about 25 to 30 degrees Celsius. Its origin is Brazil and is an exotic species. Next, we will introduce the absorption function of Econia crescens. It uses root hairs to absorb heavy metal ions and reduce the iron content in the water. The propagation method of Econia crescens includes zigzagging and sexual propagation. Zigzagging happens when new plants protrude from the end of this creeping root. The petiole has a sponge-like structure inside, and its air chamber can store a large amount of air, causing it to expand like an air sac.
Now, let's introduce the effects of peanut shells. Wood fiber contains a large number of polar functional groups, and because it contains a large amount of phenolic compounds, its absorption effect is second only to activated carbon. This is the prototype we designed. We use peanut shells for absorption. The wrinkles on the surface of the peanut shells can increase the surface area. Thus, the shells do better in absorption. The float part refers to its airbag, which can float on the water. Multiple floating boats are connected in a series to simulate the growth of Econia crescens stems. The device is easy to be recycled. Moving on to the limitations of this design, we can see that the more peanut shells are used, the absorption effect decreases. Our correcting method is to use ultrasonic vibration. This physical desorption will not harm experiment subjects. The second limitation is that this device can't be applied. Therefore, we think that the initial design can be used as the prototype for device variations, which can be used in different water bodies, such as closed and open water bodies. Finally, our conclusions are as follows. First, some details still need to be adjusted before our device can be actually used in environmental protection. Second, our device can effectively solve the pollution of nitrogen-containing waste and heavy metals, thus creating more recycled water. Third, for the time being, since our device isn't perfect yet, applying conventional wastewater treatments may still be the best solution. These are the references. These are the figure sources. This is our report. Thank you.
Thank you for such a wonderful presentation. And this is great, very scientific, very well done. Thank you very much and congratulations for such a great presentation. Thank you. Can we have the fourth one from the Taiwan?
The fourth presentation from Taiwan is from Taipei Wangfang High School, and their research topic is investigation and analysis of water quality in Jinglong Park, Taipei City. And I just have to.
No.
Okay. Hello everyone. I'm Penny Wang from Taipei Wangang High School in Taiwan. Today, I would like to show you about water quality evaluation in analysis conducted in Xinglong Park in Taipei City. Okay. The reason why I want to undertake in project in this role in my initial speculation with a survey in the sky. Therefore, starting anew, I began automated observation with my teacher. During this period, we not only record change in health, but also quantitative factors such as soil temperature and rainfall. One day, I discovered the dike roof by in a rain change, and upon minute, I found the pH low in the water and become acidic. This part in my curiosity about water pollution in the water source to affect water quality and caused me to devote into the underlying reason behind this phenomenon. So the base but the reason in analysis of pollution source our our experiment in single part in Winston district divided into parts. The first part involves the analysis from common data such as dissolved oxygen level, pH, visuals, and turbidity and temperatures using observation material provided by the global program. And the second part for that focus on bitter water sample to detect the presence for several flavor. Although this is not quite enough material provided by the global program itself, it follows the protocols outlined by the program. Okay. So while rebuilding, also have been similar studies conducted in Taiwan. Researcher Chen Yuan is discussed biological indicators as the restoration for a sixth water quality in the paper and indication. Any study titled Research on the Formula for Content Result was the quality in China City, Shandong High School. Researcher Li Shending and releasing data from the environment product enumeration. So for resort, the core essence for this paper, I saw the keyword like water quality or similar low stable on the global wetland IVSS page and discover the reads late literature authors have undertaken similar rates and discussions contribute to the literature reviews. Okay. So, the first topic devote into the study comparing the fish habitat in White Dish and Maumee River. The review that High Tree located behind NSTC has summarized the linear water quality for fish supposed by water no converted in pH, visual temperatures, and turbidity. The research concludes that white fish provide a better habitat for fish in the Maumee River, and reserving how other present data and present the educate in favor the same correct nature in different locations can also convey in stage for comparison. From the comparison for water quality in natural pole found by some district in Empire district can prevent it from the quality for water. The average for each province can be correlated to the extreme, the overall concentration for ecological pole. And after completing their research, how could I preside my own study? First, let me introduce the treatment. The card in the buffalo light corner is you measure the stability from equine, including the green for pollution, and remove the bag from spicy dark icon sticker. Add the sticker on the inside bottom for white jar. Position in sticker slightly for thinner. Fill the jar into the stability line. Leg on table and pull the stability chart on the top edge for the jar. Looking down into the jar, compare the variance for the starting disk icon on the jar to the chart. The whole record the zoo as stability in JTU. And in the top corner is a tour from magazine dissolve oxygen level and record the temperature for the water sample and suction the similar light into the water sample. Until from remove the valve from water sample, keep the valve full in the top, and drop to dissolve oxygen status into the valve. Water will overflow when the top is on empty. We'll stick the top out and the vessel. No more water overflow as top in cylinders. Make sure no bubble
are present in the sample. So in top corner, a reduction from the missing pH level. And fill it into 10 ml with a water sample. Add a pH range at top and mix by invented until the pH has its 10 degree. This metacone may remain in the sample. Compare the color to sample to the pH color. Okay, record the fluid as pH. So in the low right corner is used for collecting the last low vapor. The tools covered in simple water are collected, and using casting paper, the water is compressed in the mixing cup. The substance film in the test paper are upstream and compared to the substance present in the water. Tell them the real upstream about the nature for the substance are made based on their shape, color, and size. And discussion and negative are then extensive based on observation and just like this. And recent broad the research location. The study conducted in the Asian Pacific region, the Pacific in Taiwan, Taipei City, more than resort in the Wenshan district. The resort in the Xinglong Park located a vision from Wanfang High School and academics in Roseville. And here, let's stop for a moment and talk about why we changed Xinglong Park, the closest and most convenient place to explore from Wanfang High School, so we picked it. If the change in the future, we might explore from research in will as the geographical part in different areas from Taipei City. And we had divided in the geographical part in Xinglong Park into 6 sample points for measurement and characterization include pH level, temperature, stability, and dissolved oxygen level. The results are present in the table below. Okay, so let's take inside the identification for cellular flavor. Just the limited experiment in a time. We conduct only location 6 and 3. And we calculate the result in 4 types when roll for hair, air drill, semi-roll flavor, and not sure, but it should be semi-roll flavor. Before us that location 6, we will first instant for plain roll and or hair. Is happy brown or black color. If there are plain roll
They will have more quick cluster-like appearance, leading to conclude they are more similar to here. Okay, and that is not construed exception case for now, as says green. However, if it as the real color, it possibly brings several flavors. My judgment is that appear as a short string line. If there's similar cellulose type fiber, it may be long or as a twiggy appear. However, has essence for green color doesn't rule that similar flavor. I believe that twig appear and observation shape thing man will be more processed. You can see the similar stick that allow the cellulose flavor are brown, orange, and black, and could not resemble plant fruit or hair. And lastly, there are substances that I'm sure about. It don't think resemble plant root or hair. If we consider that the rose flavor as relatively natural substance with due to late of things appear, we can actively consider that even not the rose flavor. And the three instance was plant root or hair in water sample 3. Although nothing will observe, I like to share this interesting observation with everyone. I found a long, vibrant green margin to remarkable similar in appear to Samuel's flavor. That means to make mistake in judgment frequently. And while I am most certain that water sample 3 considered cellulose flavor, although the color is very light, it is having a thick appearance when I increase the saturation for excitement result. Imagine use photo edit software. It becomes apparent that both will distinctly red. Now let's draw prominent exo contingent based on all settings so far. And the stability in point 1 is 100 JTU, and the stability for the other 5 points are far between 13 and 14. It can be seen that the stability for point 1 is higher than the other points. Location 4 is natural pond water circulation department, and 3. The temperature in location 4 is 13.9, and the temperature in the other 5 points are fall between 18.3 and 18.9. It can be seen that the temperature at location 4 is lower than the other points. The pH voltage in creation
5 is 8.5, and the pH values for other 5 points are fall between 7 and 7.8. It can be seen that stability for location 1 higher than other points, and stability in location 1 is 100 JTU, which is higher than other points. The water depth would change to location 1, which make it easier to install the tank below the collecting water sample. Next to collection 1 is the wash basin, and the church may flow erratic into archaeological hole. And location 4 is natural pond water circulation enforcement. It's important part in water conservation system, so the rate circle and Midland or relative residual. The temperature at location 4 is 17.9, which is lower than other point. Is this less the temperature is lower because the water is coming in the time in all the time. So for the pH value in location 4 is 8.9. 5 is relatively non-falling water area into the hole, so this help him an environment with more algae growth, algae restoration, and photosynthesis lead to the more alkaline distribution. Thank you for your listening.
Thank you very much for a very elaborate and extensive presentation. This is wonderful, very scientific. Congratulations for such a great presentation. Can we have the 5th presentation from Taiwan? And I request participants also to take to take care for the time also. This is the 5th presentation from Taiwan. This is the 5th presentation.
No, this is the last
No,
one
this-
from Taiwan, 6th presentation. Yes.
Okay.
This one. Hello everyone, we are Xindian Junior High School in Taiwan. Our title is Rhinogobius yamisenensis. How does the Rhinogobius look about? It looks about 3 centimeters. It's small and middle-sized fish. It has pyrotal fins two dorsal fins, caudal fins, and anal fins. Sometimes the anal fins will become the suction's cup. Where to find the rhinogobius? We found in typical to temperate regions in East Asia. And where can we find Rhinogobius yamisinensis? This species can be found in the mid to low reaches of streams in North Taiwan, or occasionally in tributary and slow flow area at low altitude. Let's check on the picture on the right side. The red points indicate the distribution of Rhinogobius in North Taiwan. Check. The habitat of Rhinogobius yamisinensis is very extensive. It can be found in various habitats, whether it's upstream, midstream, downstream in rivers, or even in mountain streams. In the food chain, medium to large-sized fish, turtle, and water bird prey on Rhinogobians, while Rhinogobians feed on freshwater shrimp and aquatic insects.
This photo of Rhinogobius yamisinensis collected from different streams. You can see that there are differences in appearance between them. I think the reason for such differences is because they see fish that cannot migrate, so the has been no cannot exchange between different groups for a long time. However, during the process of collecting in the field, I also discovered some special phenomena. The color of these bars tended to change with the similarity and the outlet of the water. For example, the bar color of Rhinogobius yamisinensis in a stream is more acid with the black and more yellow. And in a stream it is more neutral. The color of its part will be dark and red, but since I have not yet started experiment to verify. In terms of body color, it will be affected by the degree of appetite at moment. In an open environment, the body color of the Rhinogobius yamisinensis is dark and very dark. In a potential environment, the body color of the Rhinogobius yamisinensis is black and appears black. I liking its change is due to the environment is under great passion from predators, so a dark body color is needed to form a protect through my point. I also the expression environmentation the follow is the environment method. This is expression moment method. As you can see that. After a week, it can be felt that for both male and female, the body color of those dressed in time environment is dark, while the body color of those dressed in a light environment is light. There is no change in the body color of the critical habitat environment for less rhinogobius. Sensors will change body color depending on the environment.
Through on-site observation, we have determined that the habitat water quality of Rhinogobius yamisanensis varies significant in terms of acidity and alkalinity, spanning two geological regions, volcanic rock area and sandstone shale area. Generally, the water quality in volcanic rock area tend to be more acidic compared to the sandstone and shale area, influenced by volcanic geological condition. This distinct geological vegetation plays a crucial role in shaping the acidity level of aquatic environment for the Rhinogobians yamisanensis. And then we want to share the discovery regarding the appearance and pH value of water in different regions. Firstly, we found the Rhinogobians yamisanensis when living in alkaline water, supposedly has a higher abundance of spots on their bodies compared to their counterparts in acidic water. This is quite interesting as it suggests that water quality may have an impact on their appearance. Furthermore, though field observation and sample collection, we discovered that the Rhinogobians yamisanensis in sandstone and shale area has a deeper coloration compared to those in volcanic rock area. This may indicate their adaptability to different geological features. Different place relative body colors and habits. According to the Rhinogobius research, due to the Rhinogobius didn't have mobility, different places in the environment to be short of DNA to follow. These two different places have different phenotypes. Moreover, environment of habits for rhinogobius, but these colors have effect.
Hello? Hello?
Hello.
Can you hear me?
Yeah, I don't
Now
know.
we have how many?
We
12
can't hear
people.
from them.
8 from Thailand, 3 from Philippines, and 1 from Nepal.
Yep.
So what I suggest is that if we can have a 5 minutes, just 5 minutes break, not more than that, just 5 minutes break, and we start with the presentations from Thailand, then Philippines, and the last one is Nepal. Indian participants note has yet come. So is it okay?
Okay.
Okay. So
Just 5 minutes break, not more than that, because we have 12 more presentations. All the presenters from Thailand, they are here, 8 of them. Thailand group.
yeah, I think everybody is here from Thailand and Philippines and Nepal also.
Okay. So we have 12 more, just 5 minutes break, not more than that. Just a technical break for 5 minutes. Okay.
So I'm putting a timer here so that everybody knows the 5-minute break.
Okay. Any question, any suggestion in the meantime, any point you want to raise? Chitrakar, Road, you want to say something, anything you have in mind? Road?
अभी कितनी देर चलेगी?
अरे कितना टाइम है? 2 घंटे हो गए।
तो कितनी देर तक है?
ये चलेगा 2 बजे तक।
Hello, everyone. Welcome back after the break. And we will resume with Globe Thailand. And the first presentation from Thailand is from Chomchong Ban Nimbain School. And the research topic is comparison of soil properties and abundance under various agriculture utilization.
Hello, sir. We are representatives from Thailand who will come to present our research on wetlands. We are studying in second at Chumchon Banamdip School. Welcome to our presentation.
Okay, I will share the video clip.
Yeah. Okay.
Hello, committee member. First, I would like to introduce myself. My name is Napaporn. My name is Dumuthai. And me, Supasita. We are from Chumchon Banamdip School, Thailand. Now we are going to present about comparison of soil, property and fertility under various agriculture utilization, a case study of Nam Yuam Riverside, Nam Dip Village, Mae Yuam Sub-District, Mae Sariang District, Mae Hong Son Province. Next. Let me start with origin and importance. Soil is an important resource for plants and life on Earth. If the soil isn't abundant or physical property unsuitable for planting, the plant will not be able to grow next. Nam Dip village located on Mae Yom Sub-District is flat area next to the Yuan River. Farmer farm the land without suitability lead to soil degradation, so the soil quality evaluation is more important for farmer to plant the appropriate agricultural land when they farm each type of plants, including to adjust soil quality and increase the produce efficiency. Next, our team is interested in the study of soil property under the different agricultural systems to compare the results, solve problems, preserve the natural resources and local environment. Moreover, to share the best method for farmers. Next, research questions soil property and soil abundance on the various agricultural land at the flat area next to the Yuan River, Mae Yom Sub-District, Mae Sariang District, Mae Hong Son Province. Are there any different? How? Research hypothesis. There are any different. The soil property and soil abundance on the various agricultural land at the flat area next to the Yuan River. Next. Then I would like to tell you about research objective. First, to study physical and chemical of soil property, the soil abundance on the various agricultural land at the flat area next to the Yuan River. And the second, to compare the soil abundance on the various agricultural land at the flat area next to the Yuan River. Next. Research scope, agricultural land, Mae Yom Riverside, Nam Dip Village, location, latitude 18,1437 knots, longitude 97,92543 east. Next, expect outcome. First, to know about the physical, chemical, and abundance of soil on the various agricultural land. Second, get the fact to resolve and preserve the soil quality, and the last, to share and discuss about the resource to others such as school, village, farmer, and people who interested in this project. Next, operation research, determination of the study location, agricultural land, Mae Yuam Riverside, Nam Dip Village, Mae Yuam Sub-District, Mae Hong Son Province. There are 70 support. Next, there are 5 steps to the research process. Step 1. Survey the agricultural land, Mae Yuam Riverside, Nam Dip Village. Survey the agricultural land, Mae Yuam Riverside, cultivate the plots and create a diagram of the area. Next, part 2. To survey the type of plant and farmer management in the sample area. To survey and ask about the type of plant in the plot agriculture area. 14 sample farmland are as follows. Take 2 of each. The most farmland are 4 pens, 2 plots of empty area and fresh area. Are the data about farmer plot management. Next. Part 3. To study the physical property of soil. Take a soil sample by growth methods. To dig a depth of 30 cm from the surface. To study soil structure by growth methods. Take soil color by growth methods. Use Munchell color system. Take soil consistent by growth methods. Classification of soil taken by growth methods. Next. Part 4. To study the chemical and the abundance of soil. pH take by growth methods. By pH method, to study the abundance of soil by growth methods. By NPK soil techniques. Next. Part 5. Analysis and research conclusion and send data into growth data action.
Next, result +1 to explore the agriculture area as the Yom River First Area, Namdip Village. Table 1 to show the study area, 70 contiguous area. This land map shows you about the contiguous area according to location coordination. Next. Part 2 to explore the type of plants in the agriculture area as the Yom River First Area. This table shows you about the several results of the types of plants or research area. The most plants on this area are as follows: rice, lettuce, and water spinach. And the less plants are as follows: Sylvania, grandiflora, and acacia. 14 simple farmlands are as follows. First, take 2 of each. The most farmland are as follows: rice, lettuce, water spinach, Santo, and cucumber. Second,
No, look outside, Faye.
are as follows: barren and flat area. Next, this bar chart shows about the farmer vegetable plot management to farm the various type of plants in simple area. The first bar shows you about plants the farmer can type of at 78.57%. The circle bar shows you about the vegetable plot management. The farm no part of the over S5714. The third bar shows you about S857. The last bar shows you about the insecticides. The farmer uses insecticide S8571. Next. Part 3. To study the physical property of soil in the study area. Table 3 to show you about all of pots has the granular soil structure in the study area. We found 3 colors of soil were as follows: 11 brown, 2 dark yellowish brown, and 1 dark grayish brown. Soil consistent soil was variable. The classifier of soil are as follows: S of the nickel and X and 6 of the nickel. Next. Part 4. To study chemical property of soil and the soil fertility. Table show the pH analysis and soil fertility. All pots have the natural pH. Nitrogen quantity 1 medium, 3 low, and 10 track. Phosphorus quantity 5 low and 9 track. Potassium quantity 1 high, 4 medium, 2 low, and 7 track. Next. Conclusion and discussion. Random study of 14 pots. Analyze and compare soil physical property and soil abundance. Pot 12 is abundant and has 3 types of nutrients. The Land Development Department has provided data as the lowland area was flooded. Then will be sediment accumulation, cost organic matter and nutrients. When flooding, the pH varying are shallow and low, but it also have 3 types of index pot. Next. To find the type nutrients in the pot 9 and 14. Pot 9 has the oil of plants, which is cucumber and leaf the empty area during on October.
The physical property are sandy clay soil structure is round and dark yellow with brown is a loose soil and volume pH is 6.89 in the soil was found low level of nitrogen, medium one level of potassium, and didn't find the phosphorus. Also, the plant needs less phosphorus than nitrogen, but stance is regularly. The phosphorus is necessary for the growth of roots, stem, leaf, flower, and fruit. Soil pellets hold phosphorus well but are easily lost with its data about nutrients. List of cucumber by authors of Nipon Chayamongkol. So in the pot 9 was found to be mixed with sand. The soil texture group has an ability to hold water and absorb a lot of nutrients. Soil mixed with sand can easily lose phosphorus. Next, part 14, pest management. There are many wet areas on May to September. Then on October, the farmer fruit and plant corn. Physiology, this pot is sandy clay soil structure, this granula and dark gaze brown. pH analysis, this pot has pH 7.18. Low phosphorus, medium potassium, don't have nitrogen. Next, due to the comment about the fertilizing for economic field crop from agriculture production side research and development division that show about the effect of corn by a significant low of nitrogen in the farmer study until go of the seed generation periods, especially blooming periods. Nitrogen much more important for the primary periods of corn growing because corn absorbs nitrogen and use it to build the common parts of plant. The large farmer crops them and removes them from farmland cause the nitrogen loss. Next, conclusion of plot. The most fertile soil doesn't have cropping plants and they were flat. Next, there were some plots that all year plant by only one type of plant took some fertilizer and insecticides. Next, 11 plots different from other by type of plant and soil management. The reason of soil property and soil fertility comparing under the agriculture to show the effect of the relationship between the plot management, the fertilizing and insecticiding, and soil fertility at farmland. So these reasons are important for farmers because they realize this important factor in soil and they can apply the appropriate soil and sustainable agriculture. Next, suggestion: the evaluation of soil fertility with reference to the nutrient quantity to evaluate from the analysis of plant or product and thoroughly soil and this research should analyze by other methods. Thank you for your kind attention.
Hello? Hello? Okay, this is wonderful presentation on soil. This is great, thanks. And congratulations. Can we have the second presentation? Because I have to run up because there are 8 presentations from Thailand. And they make very good presentations. Thank you.
So the next presentation is from Princess Chubabon Science High School, Trang, and their research topic is a study of soil type affecting the growth and survival rate of Iluana's acrirodes.
Hello, my name is Baramit Prayutpong.
My name is Nathavat Suntriwong.
My name is Gidipop Yim Yong.
Today we will present our project in the title of
Soil type affecting survival and growth rate of broken enhanced acoroi wash on shore. In this project, we will call Inhalasacchoroid as seagrass because they are type of seagrass. Well, seagrass is a very important plant for the ecosystem and is also food for many aquatic animals, such as dugong and turtle. Because the amount of seagrass has decreased continuously and is also caused decreasing of dugong. One of the important factors to lose seagrass is that they were blown along the shore by waves. However, in Trang, Thailand also has many natural sites of seagrass like Rajamankala Beach, Bunkong Bay, and Makam Bay. And we also have exactly soil that they grown in nature from those natural sites. So these are the important reasons that why we would like to study soil type affecting the growth and survival rate of seagrass. Research question. Does each type of soil affect the growth of seagrass differently? Second, what type of soil is the most suitable for nursing damaged seagrass? And the last one is, after releasing recovered seedling into the sea, does nursing seedling establish as good as normal seedling?
Material method as shown in the picture. This is our study site. We choose 3 study sites for studying, namely Rajamankala Beach, Makam Bay, and Bunkong Bay. Step 1. Soil samples were collected from 3 sites. Analyze soil quality according to the growth method. We also check soil texture, soil fertility, and pH. Step 2. First, we prepare the 12 aquarium box for a soil type and a soil type have for replication. Then install oxygen booster, push soil sample in the box, add seawater. Second, we prepare seagrass, trim away the rotten parts and cut off the branch. Step 3. First, we plant for seagrass seedling in an aquarium box. Put them facing the same direction in open area. Second, we measure leaf length, number of leaves, and control the salinity. Step 4, we shoot Bunkong Bay for field planting and plant 40, seeding in 9 square meter for each type of the grass seeding and then after survival rate after 1 month of field planting.
This is our result from the seagrass nursing experiment in tree soil from the study site. Found that significantly different between the range of leaf in tree soil types, and soil from Makam Bay give good result in growth rate, followed by the soil from Rajamankala Beach and Bunkong Bay, respectively, show at figure 1. Next. The width of leaf was not significantly different from nursing in tree soil type. Next. Counting number of leaves found that the seagrass nurse in the soil from Makam Bay had the highest leaf number, shown at figure 3. According to the study of seagrass nurse leaf, it was found that the seagrass in the soil from Makam Bay had the best growth rate. However, if we consider the soil quality in figure 4 and table 1, it is found that the soil in Makam Bay has the highest amounts of organic matter. Next. From study found that nurse seagrass planting at Bunkong Bay has survival rate higher than seeded seagrass as shown in figure 5. Next. By comparing the 2 seeding production method, found that the nurse seagrass method is better than seeded seagrass method because of shorter time and get more survival rate after planting. Next. Conclusion. The growth rate the seagrass plant in the soil at Makam Bay is the best. The type of soil suitable of growth rate is sandy loam. Testing the amounts of organic matter and nutrient in the soil after the experiment found that both were decreased. survival rate of nurse seagrass and seeded seagrass after planting at Munfung Bay found that the nurse seagrass has survival rate about 3 times higher than seeded seagrass. Next.
Benefits of the study. First, we can enlarge huge numbers of seagrass seedlings needed only 3 times shorter than the traditional way. The second is more economically for replanting seagrass. And the last one is making recovering of the environment more realistically, efficiently in time to increase marine life and environment. And we would like to thank you this organization, especially Princess Julapon Science High School Trang.
Okay,
Thank you for this.
thank you for the wonderful presentation, and also congratulations. How many times I say these are all so scientific, so wonderful presentations, and I must congratulate all the Globe students and then everyone helping you for this wonderful presentation. And now we have the third presentation from Thailand.
Yes, so the next presentation is also from Princess Chuvabon Science High School, and their topic is a study of microplastic in topsoil affecting the behavior of fiddler crabs in Trang, Thailand.
Okay. Hi, everyone. I am Naripat Aisanon. And my friend is Parima Noinadi. We are from Princess Julapon Science High School, Trang, Thailand. Today, I would like to present our project, a study of microplastic in the topsoil affecting the behavior of fiddler crab, Ostuca anulipes, in Trang. One of the biggest problems that the world has to face right now is the problem of microplastic waste. Currently, there are up to 112 pieces of microplastic in 1 cubic meter of seawater, and the trend is increasing steadily. Oh, sorry. I think I share the wrong screen. Okay. Can you see my screen right now?
Yes, it shows research question. Yep.
Okay, I'm so sorry. Let's do it again. Okay. Hi, my name is Naripat Aisanon.
And my name is Parima Noinadi.
We are from Princess Julapon Science High School, Trang, Thailand. Today, we would like to present our project, a study of microplastic in the topsoil affecting the behavior of fiddler crab, Ostuca anulipes, in Trang, Thailand. One of the biggest problems that the world has to face right now is the problem of microplastic waste. Currently, there are up to 112 pieces of microplastic in 1 cubic meter of seawater, and the trend is increasing steadily. Studies have also shown that hermit crab exposed to microplastic are less likely to exhibit any behavior. And furthermore, studies found that microplastic are also get into ecosystem and human body. In the mangrove forest area, some of the waste is brought from the house into the sea. At this point, there's also a kind of animal that lives and has the first interface with soil, which is the fiddler crab. We therefore led us to the first study of the impact of benefic microplastic on the behavior of fiddler crab, Ostuca anulipes, in Trang Province, Thailand. Here's the question. First, are the amount of microplastic in the topsoil and in the fiddler crab around Ban Motanoy and Salty Hot Spring different? Second, does the microplastic in the topsoil affect the behavior of the fiddler crab? And the last one, does the presence of microplastic on the topsoil affect the behavior of fiddler crab in terms of its sex and size differently? This is our material, and this is our chemical. From the methodology part, first choose the study site. We choose two different areas. First one is the abandoned mangrove forest area, Salty Hot Spring, and another one is a fisherman community, Ban Watanoi. This is our overall methodology. First, we do the land cover and collect the soil sample and feeder crab from the study site, analyze the feeder crab behavior by recording a video clip, and analyze soil quality and microplastic in soil, analyze the microplastic in feeder crab, and after that, analyze the statistical data. And the soil quality measurement we study about three important aspects, which are pH, NPK, and organic matter by growth principle. And the procedure for microplastic analysis, we save the soil with a seal of size 5 millimeter and 1 millimeter, bake it, and use ferrous sulfate and hydrogen peroxide for the digest process, and bake again and examine the sample with a stereo microscope. We do the same process in the feeder crab, but first we have to cut the specimen into small pieces to make it easier for the digest process and use potassium hydroxide instead of ferrous sulfate. And observing the behavior of feeder crab, we record a video clip for 7 minutes each from 15 samples of each crab type to study their behavior as follows: feeding, walking, rotation, burrowing, hiding, fighting, cleaning, and standing.
At the results part for the soil quality study. In soil quality study, first is pH and NPK. In both of it, in Ban Watanoi area has more pH and NPK. In contrast with the organic matter, that Salty Hot Spring has more organic matter than Ban Watanoi area. Next is the study of microplastic in the soil and also in the feeder crab. In both of this sample, we found the same, like in Ban Watanoi area has number of microplastic more, and the most common size of microplastic is between 1 millimeter to 300 micrometers. And next is the number of microplastic in feeder crab in each sex. In each sex, we will see that in female will detect more microplastic than male feeder crabs. And from the same table, we will see that in the big feeder crab will detect more microplastic than the smaller one. And this is the characteristic of microplastic that we found. We found only two types of microplastic, which is fragment and fiber, and found fiber. This is the example of microplastic that we found. Next. Next is the characteristic of microplastic in their colors. We found many colors of microplastic, which is clear, white, red, black, green, and blue. And this may be come from a fishing net. And this is the example of microplastic that we found. Next is the study of the behavior of feeder crab. First is in the figure 3, it shows the average behavior changing in 7 minutes. You will see that the feeder crab in Salty Hot Spring area has more changing behavior more than the feeder crab in Ban Watanoi area. And the figure 4 shows the feeding rate of feeder crab. Is we'll see that in Salty Hot Spring area has faster time to feed their food than the feeder crab in Ban Watanoi area, and the female will eat faster than male, and the smaller one will eat faster than the bigger one. Next is the original flaw of feeder crab. In this study, we didn't see any difference in this. And next is the study on the behavior of feeder crab over a period of 7 minutes. First is the behavior group that we found in male feeder crab only, with its predation and fighting behavior. In both of these behavior, Ban Watanoi feeder crab has time to predation and fighting more than Salty Hot Spring feeder crabs. Next is the behavior group that we found in Salty Hot Spring area only, which is hiding, burrowing, and cleaning behavior. And next is the behavior group that we found in both study sites, which is feeding, standing, and walking. In feeding and standing, Ban Watanoi area has time to feeding and standing more than Salty Hot Spring area, contrast with walking behavior that Salty Hot Spring area walking more. And in the conclusion, microplastic in the soils, in Bamatonoy areas higher than salty hot spring area for 5 times, and the discourage was blue, green, black, red, and white, and this maybe come from a fishing net, and the microplastic in the feeder crab is also the same, like in Bamatonoy area has higher number of microplastic than salty hot spring area for 3 times, and the more microplastic detect female more than male, and the bigger feeder crab, the more microplastic accumulated. And the feeder crab behavior. Microplastic may be entered in the feeder crab and affect their metabolisms. Like from the result, the feeder crab are to the feeder crab in salty hot spring that noticeable from walking less, non-burrowing, non-hiding, non-cleaning, more standing, more feeding, but slower feeding rate, contrast with the feeder crab in salty hot spring area, which actively and faster. After we finished the project, we went to the area to expand the knowledge that we know to the community and the school that under care of our school, Bamatonoy School, and let the children know about the danger of microplastic waste and the benefit that can be gained from take caring the area to increase the value of tourist attraction. And we would like to thank to our supporter. And this is our reference. Thank you for listening. ขอบคุณค่ะ
Thank you ขอบคุณครับ
Thank you for the wonderful presentation, the microplastic in soil, which is a very new topic, and I am sure that this will be taken up by several schools in different countries, and this will become one of the activity which we think we can have in 2024 and 25 as a globe regional activity. Thank you very much for such a wonderful presentation. Thank you. Can we have the next presentation?
Yes, the next presentation is also from Princess Chubavan Science High School, and their research topic is innovative mosquito OV traps for reducing mosquito outbreaks.
Hello, we are from Princess Chubavan Science High School. My name is Piyapat Phedhin.
My name is Kanakon Hongsun.
Today we will present our project is innovative mosquito OV trap for reducing mosquito outbreak. And then while we're doing that, because from the WHO report show that a spread of mosquito-borne disease is still high in Thailand and tropical countries. And in the past few years, a pandemic of COVID-19 situation that makes the most people stay at home, make more activity to do while at home, including planting tea is an activity that many people are interested to doing that. So we aim to create mosquito trap template to reducing the number of mosquitoes by using the SketchUp program to design a prototype and print the workpiece with the 3D printer. And next, come to the objective. The first one is the innovation create and develop can intercept, trap, or lure mosquito to lay eggs. And the last one is to study the mosquito breeding container to have the most mosquito laying eggs in order to be used as a model for creating innovation. And next, we're presenting you engineering design process that has 6 steps. To begin with, we collect information and ideas to solve the problem. After that, we design or develop innovation, and also we test the innovation to see if there is any flaw. If yes, we then jump to modification and improvement to increase the efficiency of work of this innovation. The improvement process are done for 3 times until we get the best version of innovation. We finally present innovation. And I will explain the process, how we're doing that is step by step. Begin with the first step, problem identification. Our study site where we collect mosquito data from covering 2 districts, Amang district and Hasaran district, totaling 24 households. And next, we go to the study site to explore mosquito data and classify the type of mosquito breeding ground that we found according to the GLOBE protocol. There are three formulas that we use to calculate mosquito index, namely house index, container index, and ritual index. And come to the result, there are 24 households where we collect mosquito data from. It is found that 163 breeding sites are found within 20 houses, and only 172 breeding sites found mosquito larvae. Specifically, Aedes mosquito larvae are found in 102 breeding sites out of 172. And the pass on mosquito index. The mosquito index revealed that Trang province is at a very high risk of dengue fever. And this slide shows the sample of breeding site that we found. It was found that the plant pot resources was the container of the mosquito breeding source at 40.16%. So we aim to create mosquito trap plant pot that can plant tree, meanwhile can trap mosquito in the same time, that can be used instead of traditional plant pot.
Into the related information search. Normally, when we water the tree, water from watering will be stuck at a saucer. That is the reason why the pot become a breeding site for mosquito. So we use water from pot saucer to attract mosquito. The factor that lure mosquito to lay egg is black color and humidity, because the mosquito like to lay their egg in the dark and the high humidity. And this is the mosquito trap that had before, which will be adapted with our innovation. And let's see how we create the innovation. We created and developed the mosquito trap plant for 3 versions. In version 1, by designing the shape of the mosquito trap is a cylindrical shape. It had 2 parts. The upper part which is used to plant the tree, the lower part for mosquito trapping. There had a hole for mosquito to fly inside. In this version, made from the plastic bottle and plastic bag, and we found that first, there are a few mosquitoes in the trap. The second, they can trap adult mosquito, and the last one is the material used is weak. So we developed it into version 2. In this version, it still had 2 parts similar to version 1, but we increased the number of holes into the mosquito trap to allow more mosquito to fly in the pot. We used a 3D printer, created a pot to strengthen it, and we designed by using the SketchUp program, and we found that first, an adult mosquito can fly out. Second, soil and sediment fall into the water, and it may also be a breeding ground for other mosquitoes. And the last one is mosquito egg that fall into the water. When grow as adult, they're able to fly away. And the last one in version 3, we designed the hole to have a wide at the top and narrow base. It's like a cone. We added the net to protect the mosquito escaping, and this is our principle. When we watering the tree, the water will flow down at the bottom of the pot, and the high humidity and the dark color will lure the mosquito that they come to lay egg, and the egg will flow down to the bottom. And the mosquito that cannot escape because our net protection, and the egg will grow up to the adult stage, and they cannot escape. Then we bring out 3 versions of the pot to test and compare their efficiency. From the result of the study, the third version of the mosquito trapping plant pot was able to catch the mosquito. And you can see that the mosquito trap plant pot version 3 is more effective in trapping mosquito larvae, pupa, and adult than version 1 for 1.77, 3.5, and 1.5 times respectively. In conclusion, the plant pot, which are the most common breeding site, has been used as a model for creating and developing mosquito trap plant pot. And the last one is the mosquito trap plant pots had been developed in 3 versions. The mosquito trap plant pot version 3 is more effective in trapping mosquito larvae, pupa and adult, then version 1 for 1.77, 3.5, and 1.5 times respectively. It can be seen that our mosquito trap plant pot can actually be used in our innovation, can replace ordinary pots and can be used to reduce the number of mosquitoes and reduce the risk of dengue fever. And we would like to thank our supporters. This is our reference. And thank you for your kind attention.
Hello, thank you very much for a wonderful presentation. And I must congratulate the presenter for such a wonderful presentation. And maybe we can have next presentation from Thailand, which will be third one. Yeah.
No, this next one will be the fourth one, and that is from Matthew Comark School, and their topic is comparison of soil quality and yield of oil palm planted in shrimp paddy soil and peat oil in Nuam Nangkham district, Patong Lang province. Okay, let me change.
สวัสดีครับ Good afternoon, ladies and gentlemen. First, I would like to introduce my team. My name is Namdal Lakman.
My name is Sakia Liso. My name is Vida Liso.
We are from Madhyam Comark School. A present day to everyone. Today we are going to present about our project, comparison of soil quality and yield of oil palm plant and shrimp paddy soil and peat oil in Got Nangkham, Pavilion District, Province.
National and background. Got Nangkham sub-district area Papayun district, Patong province is a knowledge part of Songkhla Lake. Most of the area is swamp forest. Farmers used to ride going giant tiger ponds.
Shrimp farming is currently being impacted by price drop and production cost and high, causing products to lose money because of the price increase in oil palm. Farmers in most areas are turning to everything. It has been difficult to grow corn for a long time since the salt and sodium contents of the soil are higher than usual due to the use of chemicals.
ขอโทษค่ะนักเรียนPowerPoint นักเรียนไม่ขยับเลยค่ะ ขยับนิดนึงได้ไหมคะ
โอเคค่ะ
แป๊บหนึ่งนะครับ
โอเคค่ะ
The salinity of seawater and the extensive number of ponds where there cause soil erosion and are unsustainable.
Research question. First, is there a difference in soil quality between oil palm plantation plants on shrimp palm soil and peat soil? Second, is the yield of oil palm plants in shrimp farm soil and peat soil different?
Research hypothesis. First, the soil quality of oil palm plantation developed on peat soil is superior to the soil quality of shrimp farm soil. Second, the yield of oil palm plant on peat soil is higher than that of oil palm cultivars is in shrimp farm soil.
Research objective. First, to compare and contrast the soil quality in oil palm plantation plants on shrimp farm soil and peat soil. Second, compare the oil palm yield in shrimp field and peat land. Third, provide information on soil quality development to local farmers.
Impact benefits. First, let's learn about the soil quality in oil palm plantation, shrimp farm, and peat land. Second, learn about the output of oil palm plant in shrimp farm
on peatland. Third, provide local farmers with knowledge to help them improve soil quality.
Scope of the study. First, study area. This study area list picture. Oil palm plantation area sets up in shrimp palm soil. Study area live picture. Oil palm fence on peatland.
Task 1. A comparison of soil quality in oil palm plantation plants on shrimp farm soil and peat soil. First, let's getting soil sample for physical and chemical analysis. First, measure the size of the study area. Second, choose 5 subplots per study area as shown in picture 7, which is subplot size 20 multiplied 20 meter. Third, select the soil sample which in the bushes 3 plants per subplot by digging the soil in a wedge from about 30 cm deep inside the bushes at 3 spots, front, left, and right.
Collecting soil sample to the method by GoPro protocol.
Second, soil quality analysis. Follow the instruction provided by GoPro. Soil quality, physical characteristic, soil structure, soil solidification, soil texture, soil moisture, chemical property, using an LAD, soil kit, soil pH, soil fertility, nitrogen, phosphorus, potassium.
Task 2. A comparison of oil palm productivity. Go on shrimp farm soil and peat soil. First, visit each location to weight the oil palm bushes on the day of harvest by choosing the tree from which soil sample were taken. Second, record the weight of palm bushes on the data recording from. Third, compare oil palm results in both locations.
Result according to table first. The soil structure, condition, and texture of soil from oil palm plantation that's used to be shrimp farm and peat forest are different.
Image first show that oil palm field go on peat soil have higher soil moisture level. Oil palm plantation have go up on shrimp farm. The soil of oil palm plantation plants on peat soil and shrimp farm soil has soil moisture average value were 0.36 and 0.26 respectively.
According to image second, the average pH value of oil palm plantation cultivated on shrimp farm soil and peat soil was 5.73 and 5.27 respectively. The soil pH in both locations ranges from 4 to 6, which is excellent for growing Surat Thani to oil palm.
According to table 2, the soil of oil palm plantation plants in peat soil has a lower quantity of important plant nutrients than oil palm plantation growing shrimp farm soils, has medium to high amount of nitrogen, very high level of phosphorus, and low level of potassium.
From picture third, it's what furthers oil palm grow in peat soil gives better yield than oil palm grow in shrimp palm soil. Oil palm grow in peat soil and shrimp palm soil yield with average weight of 72.10 and 12.95 kg per bush respectively.
Discussion and conclusion. Peat soil is preferable to shrimp soil for growing Surat Thani to oil palm. Peat soil has soil structure and soil retention capability. The soil texture and richness are better than in shrimp farm, but the soil acidity and unfertility are similar.
However, Fish soil is more productive and source for growing Surat Thani to oil palm. The oil palm yield has been determined to be oil palm plants on flatlands. Products more and provide a heavy yield on average than those grown on chim soil. According by assumption 72.10 kilogram/watches. Thank you for your attention. It's what a pleasure being here today.
Thank
Thank
you.
you. Okay.
Hello.
Yes.
Again, another wonderful presentation from Thailand. Congratulations, guys, for such a wonderful presentation. And can we have the next presentation? Then
Yeah. So
this is the fourth one.
yeah, this is from Kuru Prashashan School in Thailand, and their research topic is application of environmental factor to the development of hatcher high of small shrimp in stagnant water.
This is number 4.
I think this is number 5.
Okay.
Yeah.
Okay, please.
Is number 6 from Thailand.
Number 6.
Okay, yeah. So we have 2 more. The one more after this one. No,
Okay.
2 more. Oh, yeah. Which one?
Hello, I am Thong Thai Rak Sri Suk.
I am Sushawdi Kasuk. I am Bemika Kom Iang.
We study in grade 12 at Kru Prachasan School in Thailand. We are going to present our research about application of environmental factor to the development of hatchery for small shrimps in stagnant water. Shrimp is a source of food for rural people. When the environment for water source change, the quantity of shrimps decreases. Researchers are therefore interested in studying the issue of environmental factors, the effect the spouting of shrimp in order to create an environment suitable for the spouting of shrimps in order to create the quantity of shrimp. About research question, first, temperature, pH, TDS, water quality, and dissolved oxygen in the water is the amount of water in the pond related to the amount of spouting shrimp or not. Second, can the develop hatchery to increase spouting shrimp? Hypothesis. First, temperature, pH, TDS, water quality, and dissolved oxygen in the water in the pond is related to the spouting of shrimp. Second, the developed hatchery can increase spouting shrimp. Objective. First, to study environmental factors related to the number of shrimp eggs in the pond of Kru Prachasan School and Noi River. Second, to develop a hatchery with an environment suitable for shrimp to increase spouting shrimps in the stagnant water.
This is the scope of study their water temperature, pH and TDS, water quality and oxygen dissolved. Next, this the study area. We choose Noi River in Sankhaburi, China's province, and pond in Kru Prachasan School, China's province for the project. This slide is showing our starting point. Its location is divided into point 5 in order. About the measuring the number of shrimps. The first step, we use a 30 centimeter net and swing it back and forth 5 times. The second step, we are sorting and counting the number of chimp eggs. And the last step, we correct. About measuring the physical characteristic of water, the first we check the water temperature. Second, oxygen dissolved in water. Third, pH and TDS measurements. Fourth, measuring water clarity, and then correct. Data analysis is the relationships between temperature, pH, TDS, and water clarity, oxygen dissolved with number of spawning shrimp that were solved by Pearson correlation. This slide is show about the student results. In this step, we will find that the amount of dissolved organic substances in the water and amount of dissolved oxygen in the water had correlation coefficients of 0.960 and 0.955 respectively, which were highly related to the number of shrimp eggs. Therefore, leading to the development and study of the variety of shrimp egg house. After we use the hatchery in the pond, we would like to develop the hatchery for shrimps. This is our development. We start with the frame is made of PVC pipes. A hose carries air to the bottom. Fill hydrilla verticillate for increased oxygen and camouflage. Mosquito nets protect the shrimp from dangerous bottoms. Solar cell air pump for increasing oxygen. Buoys from plastic bottle help to stabilize the hatchery. Plastic nets for entry and exit and protect fish. This shows results of the study. The study found that area with shrimp egg house tend to increase the number of shrimps more than area without shrimp egg house. The summary: temperature, acidity, alkalinity, water quality, TDS in water, and oxygen in the water of the pond in front of Kuru Prachasan School and Noi River. In the study area is related to the number of spawning shrimps, the amount of solution in water and the amount of oxygen in the water have a very high correlation with correlation coefficient of 0.960 and 0.955 respectively. The hatchery can increase the number of spawning shrimps in 4 by 4 square meter experimental area 19.42 times higher than in the area without egg house and tend to increase. Thank you for your time and attention.
Hello. Thank you. Great presentation. Thank you very much. Now we have the last presentation from Thailand now.
No, second last. And this one is from Bandun Sharatwani School, and their research topic is study of environmental factors affecting the livelihood of mosquito at Don sub-district, Kok Thai sub-district, Krong Chai.
What I'm thinking, after having this presentation, let us have some presentation from Philippines, Nepal, and in the last we have the Thailand presentation. Is it
Sure.
okay?
Okay, I'll arrange it so that the next presentation from Bandung go.
Okay. Please.
บ้านดอนเชิญค่ะ
ไม่ค่ะ เขาให้ฟิลิปปินส์ก่อน
Oh no, you guys can, this one can go from Thailand, and after that we'll take from Philippines.
โอเค
เชิญค่ะบ้านดอน
afternoon, we are presenting facts of environmental factors affecting the livelihoods of people. Statistics of China, China province.
Hello. Can you speak a little loudly? We are not able to hear you, or at least I am not.
I am Mga elementary. Yun yung maganda sa ano kasi yung elementary.
We are from Bandar Salalat School. Patung China, China, Thailand.
Origin and important mosquito become carrier of dengue fever to people in บ้านดอนหน้าสัมปายใต้ปักธงชัยนครราชสีมา province and they used to children who were dengue fever but now it has been found that no one in the village had dengue fever or interview with stars who are village volunteers to destroy the breeding of mosquito larvae. Study about mosquito larvae. Research questions. Did environmental factors
เดี๋ยวนักเรียน PowerPoint นักเรียนไม่ขยับเลยอะค่ะ
Your slides
เลื่อน
are
เลื่อน PowerPoint นิดนึงค่ะ
ได้หรือยังคะ
ยังไม่ขยับมาเลยค่ะ
ไปไหนอันนี้เรามีค่ะ
โอเคได้แล้วค่ะ
Research questions. Did environmental factors and seasons have different effects on the living of mosquito larvae in the subdistrict and Kokthai subdistrict? Hypothesis. Environmental factors and seasons have different on the living of mosquito larvae in the subdistrict and Kokthai subdistrict. Research objectives to study environmental factors and seasons in the living of mosquito larvae in the subdistrict and Kokthai subdistrict.
Teacher School, Bandar School, Bandar subdistricts and บ้านสำไลใต้คกไทย subdistricts ปักธงชัยนครราชสีมา conditions air temperature water temperature the acidity the water light intensity and oxidants durations of study 16 June 2022 to 14 July 2022.
Scope of study using the growth protocol บ้านสำไลใต้บ้านดอนสกูลบ้านดอน Research equipment meeting to plan survey of mosquito larvae. Average air temperature. The average air temperature at Bandar School and Bandar are 30 Celsius. The average air temperature at Basamlata is 32 Celsius. Average relative humidity. The average relative humidity at Bandar School and Bandar are 91%. The average relative humidity at Basamlata is 82%. Average light intensity measurement. The average light intensity measurement at Bandar School is 1,900. The average light intensity measurement at Bandar and Basamlai Thai are 1,750. Result of classification of mosquito larvae. There were coolest mosquito at
อนมอส There were amateurs at บ้านสำลายใต้มอส There were Aedes aegypti at บ้านสำลายใต้มอส There were Aedes albopictus at บ้านสำลายใต้มอส There were pupae at บ้านสำลายใต้มอส Average water temperature measurement. The average water temperature measurement at Bandar School and Ban Samlai Thai are 27 Celsius. The average water temperature measurement at Bandar is 28 Celsius.
The average measurement of water acidity base was 7 in 3%. The average oxygen measurement value was 7 in 3%.
Average classification of mosquito larvae. There were Culex mosquito at Bandar Moss. There were Amiza at Ban Samlai Thai Moss. There were Aedes aegypti at Ban Samlai Thai Moss. There were Aedes albopictus at Ban Samlai Thai Moss. There were pupae at Bandar Moss. Summary and discussion. From a study of environmental factors affecting the life of mosquito in all 3 areas, it was found that Ban Samlai Thai has the environmental factors that affect the life of mosquito the most. Because there is overgrown grass, trees, waterlogged channels, their weather is not too hot and not too cold. Mosquitoes like hot and humid weather. Moreover, this is the raining season and 3 is clean water, which makes mosquitoes lay eggs. There is a lot of growth and propagation. Containers where mosquitoes are found include cement tubes, coconut shells, car tires, pen buckets, and basins. The containers where mosquitoes are most likely to be found are 7 brown and 3 1 black and white respectively because of mosquito-like dark colors. In the raining season, mosquitoes breed well. The temperature is not hot, not too cold, and appropriate relative humidity. Benefits expected. 1. Able to classify mosquito larvae. 2. Know what kind of disease carriers mosquitoes. 3. Campaign to eliminate mosquito larvae. Feedback. The next study should study what types of people and what types mosquito like to bite. Thank you.
Okay, thank you very much for such a wonderful project which has been implemented like on mosquitoes. Thank you again. Congratulations, the guys, for wonderful presentations which you have made. And also one thing I feel surprised sometimes, Thai is the language for Thailand. But all the presentations, they are so wonderfully done in English. And I have seen you guys doing practice and the rehearsals, taking a lot of, a lot of strain and then things like that. So I must congratulate you for this great presentations with a lot of information, data, and so and so. So now we can go to the Philippines and then Nepal and then come back to Thailand. Okay?
Yeah.
Dr. Des, can I present the next, the last from Thailand before
Okay.
go to
So you
Philippines?
on the thing is Philippine student school have closed around hour ago. So they're just waiting to go out of school. So we are taking one presentation from them and then we'll get back to Thailand.
Okay.
Okay. So the next presentation is from Philippine Science High School, Calabarzon.
Rod, you want to say some? Rod is here?
I'm not seeing Rod. Let me check.
If Rod is here, you want to say anything? Okay, go ahead.
Yeah, Francis, if you are ready.
Francis, congratulations for your working group membership. मुझे फिलीपींस की चल रही Thailand की बात. We try to wind up at 2:30 at least.
इसके बाद चलेंगे हम जरा राम को लेके.
Hello. Good afternoon, sir. Hello, am I audible?
Yeah.
Okay. Good day, everyone. As part of the Global Wetlands Symposium 2024, we are here to present our study on the pH and dissolved oxygen concentration of the Kalumpang River system of Batangas City in the Philippines. We did so by sampling water from 4 different sites as indicated in the map with the green flags marking each site. I am Samantha Grace Andan, and with me is Jacob Keenman-Tok alongside Cian Antoinette Reyes, with our teacher advisors, Sir Francis Emerlino and Sir Dexter Lorden. First, let us introduce the Kalumpang River system. The name is actually derived from the tree that grows along the banks, scientifically known as Sterculia fotida, or locally Kalumpang. It is found in northwestern Batangas City and it borders Poblacion, the city center of Batangas City. The water from the river flows to the Batangas Bay around 2 kilometers east of the Batangas port. It is also described as the Nile River of Batangas, running through the heart of the city itself. It is usually considered for the development of ecotourism of Batangas City. However, the Kalumpang River is currently classified as a Class D river by the Department of Environment and Natural Resources, or DENR. This means that it is only suitable for agricultural and industrial processes. Such classification could be attributed to the fact that disposal of garbage and waste from households and livestock, as well as untreated sewage, ends up in the river. To make the river system part of the ecotourism growth of the city, the goal of the river restoration is to achieve Class B, also known as the Recreational Water Class 1, intended for sources of water supply that require conventional treatment. In the context of the study, we aim to check the river's pH and DO concentrations by using the following materials: a wireless pH meter to detect the power of hydrogen, and a wireless optical DO sensor to determine the dissolved oxygen concentration. This equipment could be used and connected to mobile phones via Bluetooth through the SparkView mobile application. Moreover, we performed globe cloud observation protocols at each site we visited.
Okay, so for the first sampling site, it was located under the Kalumpang Bridge, which is flanked on both sides of the river by heavily populated areas of Batangas City. So underneath the Kalumpang Bridge, the dissolved oxygen concentration was found to be 6.95 milligrams per liter, while the pH level was at 7.39. Next, for our globe observations, the elevation was 2 meters above sea level, with the cloud cover as scattered with altostratus, cirrostratus, and cumulus clouds. The sky at the time was somewhat hazy, with dry ground and leaves on some trees nearby. So our observations in this site was that marine life was seen on the site. So the example of this were small fishes. However, there are also sewer ends present on the site, as this is again located in a densely populated area of Batangas City. For the second sampling site, it was located in a more rural area of Batangas City, in southern Batangas City, where the pH level was found to be around 7, and then the DO concentration was found to be 6.90 milligrams per liter. So some of our observations were that green algae was found on the banks of the river, and then some green algae was also found near a decaying plant on the riverbank. There were a number of livestock, including a goat that was seen eating grass right beside the river, and then at the time that we visited, it was dry with leaves on some trees. For a third site, it was located in southeastern Batangas City. where the pH levels were higher than the previous sites at 8.32, and the dissolved oxygen concentration was found to be around 6.92 milligrams per liter. So observations, the ground was dry with leaves on trees. The water was clearer than the previous sites. However, there was still trash flowing on the river. Furthermore, this was located near a bridge construction site and a large gym that is to be used in the future. So for our last sampling site, it was located on the mouth of the Kalumpang River near Batangas Bay. The DO concentration for this site was consistently 6.63 milligrams per liter, while the pH level was around 8.09. So the observations for this site included 3.6 meters above sea level for the elevation, and the sky was overcast with altostratus, altocumulus, cirrocumulus, cirrostratus, cumulus, and stratocumulus clouds. The sky was blue, but was somewhat hazy, and the ground was also muddy and dry, and with leaves on some trees. So here are some images of the site. As you can see, it was still somewhat dirty, even though this was located in a more rural and more remote part of Batangas City. So there was a trash and a few uprooted trees located on the shoreline. So here's a site overview of Batangas City. As you can see, 3 of the 4 sites were located further north, and site 3 was located further south, as this was located in the most remote part of Batangas City. Site 1 is located again near the city center. Site 2 was located nearby, and site 4 was at the mouth. So here are some pictures of our sampling. And then here are our results. So in comparison to compare these sites, first, site 1 and site 2 had very similar levels of pH and dissolved oxygen, with around 7 for pH and around 7 as well, or 6.8 for the dissolved oxygen. Meanwhile, the more remote sites, which were site 3 and site 4, featured more alkaline levels of pH, with more than 8 for both sites, and the dissolved oxygen being similar to the first 2 sites with around 6.8 to 7.
Graph and table presented, the dissolved oxygen concentration of the 4 different sites range from 6.6 to around 7 milligrams per liter, while the pH levels range from 7, also known as neutral pH, to 8.33, that is slightly alkaline. Compared with the set standards, the dissolved oxygen concentration levels of the river fit the water body classification SA and SD for coastal and marine waters. The DO levels are unusually high given that it is a freshwater body. This might mean that the rate of oxygen replenishment in water is greater than the utilization of oxygen, also making it satisfactory for the survival and growth of aquatic organisms. Meanwhile, its pH fits the Class A standards of fresh water, which falls within the range of 6.5 to 8.5. The water sample suggests that the river system of Kalumpang fits within the SA and SD classes, especially for its dissolved oxygen concentration. SA and SD classes, as per the Department of Environment and Natural Resources, are suitable for recreational activities such as swimming, diving, and fishing. Such improvement in the water quality in terms of pH and dissolved oxygen is important, even vital, for its development and inclusion as part of Batangas City ecotourism. For future studies on the Kalumpang River, we recommend using and testing more parameters, including its temperature, total dissolved solids, and biochemical oxygen demand to ensure a more accurate classification of the sample. The researchers hope to expand this study in the future to further analyze and monitor the water quality of the Kalumpang River. That is all. Thank you.
Thank you, Samantha and Jacob. And now we will take presentation from Thailand, Bangantonoy School, and their research topic is study soil quality effect of survival and growth of small livid mangroves.
And congratulations, Philippines, for wonderful presentation.
Okay.
Yeah, now Thailand.
เชิญเลยค่ะโรงเรียนบ้านมดตะนอยค่ะ
สวัสดีค่ะ Hello everyone My name is วรรณสาวะคง My name is พัชราอ่อนแก้ว My name is Anita สุสุ We are students from บ้านวัฒนา School กัดตัง District of Trang Province in the south of Thailand We will present about study on soil quality effect on the survival and the growth of small leaf mangrove Rhizophyra apiculata Ban Mutanoi School is in Kantang District, Trang Province. The geographic is the coast surrounding with mangrove forest, especially small leaf mangrove. The mangroves are useful and benefit in the community. They can be used as medicine, firewood, construction wood, and fishing equipment. Nowadays, the number of small leaf mangroves are dwindling. This affects ecosystem. Replanting project is currently used in order to increasing mangrove forest area, but dramatically increasing number of mangrove trees are needed to recover damaging environmental. Not enough. This experiment is trying to help and boost replanting of mangrove forest area. Research question. The first, what kind of soil is suitable for growing small leaf mangrove? The second, does quality of soil in each area affect the growth of small leaf mangrove? Research mentor study area is the area at Panmatana School, Trang Province, located at latitude 7.3073 north and longitude 99.4197 east. There are three kinds of soil were using in this experiment, namely cotton sandy soil experiment 1, semi-cotton soil experiment 2, and mangrove forest soil experiment 3. These are materials that we use in our experiment methodology. We start with the first three rectangular 1.2 by 3 meter painting plots were prepared and put soil from experiment 1, 2, 3 in each plot. The second, prepare mango pot by correcting the pot that falling down from the tree. The third, painting mango pot 8 cm deep and 200 pot per plot without watering. The fourth, counting number of leaf survival rate of 50 samples, 1 a week for 10-week period. The fifth, measure soil quality by using growth soil protocols, such as soil pH, soil element, and soil organic matter before and after planting. The sixth, send soil data to growth data entry. The seventh, analyze data using Microsoft Excel. The result of measuring the use of mangrove tea in all teapots were not different. Also, number of leaf counting was shown not different, but survival percentage of the teapots were seen different, whereas experiment 1 shown good result in survival percentage.
Hello.
This picture shows mangrove growing in 3 pots. Soil texture in experiment 1 is sandy soil, experiment 2 is loamy sandy soil, and experiment 3 is loamy soil. Soil pH in teapot is not different before and after experiment.
soil fertility before and after experiment 1, which was showing reduce of nitrogen after experiment could be due to good result in survival rate. This picture shows organic matter before and after experiment. The results are not going the same way, whereas experiment 1 and experiment 2 increase and decrease in experiment 3. This also could be the effect of seed time.
Conclusion. The experiment showed that the growth rate of on 3 types of soil growing mangrove tree in 3 pots, coastal sandy soil, semi-coastal soil, and mangrove forest soil were not different, but experiment 1, coastal sandy soil, are shown having the most growth rate among 3 experiments.
Benefits of the project. The first, no type of soil suitable to grow mangrove tea. The second, no soil fertility for planting mangrove tea. The third, apply the knowledge to use in the conservation of mangrove forest at the Mutanoy community. Finally, we would like to thank all of our supporters.
Thank you for your kind attention.
Goodbye.
Thank you for wonderful presentation. And all the presentation from Thailand are very good, high standard, look like research presentations. And thanks and congratulations all the participants making this presentation. Thank you. Now we go to Philippines. The second.
Yeah, so next presentation is from Batashan National High School, and their topic is inhabitants of wetlands and their preservation.
Road is here?
I think he is, yes.
Hello po. Can
Okay,
you
please,
see
after
my
the presentation, we will request all to. Okay. The second presentations from the Philippines. इधर बैठ जा यार, वहां से थोड़ी दिखेगा.
So, there and we all know the ones they are part of cycle.
Gabriel, I'm not able to see your screen. I don't whether it is me or somebody else. Sonal, are you able to see the screen?
Not really, not at all.
Yeah. I think, Gabriel, you have a connection issue because it's saying that you are shared screen, but we are not able to see your presentation.
Okay, po.
If you could try that again, I think that it will help.
Hold on, po. Hold on.
And I guess in the meanwhile, we can announce about the regional general because people are leaving after their presentation and I can see 50
Yeah, sure, go ahead.
are left.
Yeah. Sure, go ahead.
Yeah. So I just want to make an announcement here that we're going to have a regional general very soon. And whatever presentations the students are presenting, we would expect them to be posted on our email so that we can put up in the regional general. And if any question you have, you can mail us directly about the regional general and the presentations, how we're going to put and all. We're going to give you a proper, proper format, how you can put the paper and the presentation very soon. And then you can post us accordingly. Am I clear?
Yeah, just to add to Sonal detail, we will be sending it to the CCs because we don't have the details of every student we are listening the paper from, but we are going to soon sending it to all the CCs, and we would request them to share it to the students and schools so that we can get the information back and publish it under the regional journal.
And after we get all the material, we will send to Rod.
Yep.
Who is the editor for this journal, and after his approval, we will put it on,
On the regional website.
and also try to get some printed copies if
Yes,
anyone
of
would
course.
like to have.
Yeah.
Okay, Rod, I will send you the details, and then we want to really put up this, what do you call the wetland volume very soon.
Well, actually, today's presentation, a lot of them are very good and may even be publishable. I mean, if they have those articles ready, they could just send it to me and
Yes.
I'll have it professionally edited and published. We don't have to wait for the regional meeting. We just need.
I guess, Rod, here we have to make a format how the students can send us the articles, just send us the presentation, and we have to make a format. So let's have a meeting or something like that to finalize the format so that the students, that will be much more easier for the students to, post their papers to us.
Sure.
So
Later,
I think they have
later I'll
a-
send you the submission guidelines. We have actually prepared for it. I'll send you the submission guidelines, and that's the format that
I will give. Let us finish 3 more presentation.
Yeah.
Okay, so it seems like Gabriel is having some Wi-Fi issue. So if he doesn't mind, we can move it to the another Philippine school. In the meantime, he can sort out the network issue and send us the presentations to us so that we can share it on his behalf. And the next one is from Philippine Science High School, Central Quezon region, and their topic is impact of lab on water quality of Chinon's aquaculture in Dopang, Panangsinay. I'm sorry if I butchered the name. And yeah, if Aisha is here, she can start presenting. I think she is. Let me check if she hasn't dropped.
Hello, I'm just trying to present my screen right now. If you don't mind waiting a few minutes, a few seconds, sorry. Is my screen visible now?
It's visible.
It's okay now.
Thank you, sir. So, good day. Before proceeding with my presentation, let me introduce myself. I am Aisha Brianna S. Alemania from Philippine Science High School, Central Luzon campus in the Philippines, and I will be presenting a study I conducted with Rafael Carl P. Rosario and Leonardo M. Samana for the Globe International Virtual Science Symposium 2023 entitled Impacts of Lablab or Algal Mat on Water Quality of Chanos Aquacultures in Dagupan City, Pangasinan. For this study, we were advised by Ma'am Josephine Joy T. Antalan, and we used the Globe Hydrosphere Protocol. So first, let me define to you some terms. Dagupan is a city in Pangasinan that thrives on its local fish industry with bangus or chanos or milkfish as its specialty. It is also dubbed as having the best tasting bangus in the Philippines. Moreover, it yielded 109,893.50 metric tons of bangus in 2019. 1/4 of the entire country's total yield. Next, algal bloom is a prevalent cause of fish kill. In 2002, the coastal town of Bolinao, Pangasinan, experienced a fish kill which cost them 6 million pesos. And 2 decades later, in 2022, they experienced another which cost them a significantly larger loss of 500 million pesos this time. However, both events were also highly associated with the decreasing water quality in the area brought on by excess nutrients from synthetic fish feed. So what could be the solution to this problem? Here comes in lablab, or algae mats in milkfish ponds. It has a rich concentration of essential fatty acids, thus it is being used by milkfish farmers as a supplementary or full dietary feed for their milkfish. Thus, this study aims to determine whether lablab is beneficial to Bangos aquaculture and the environment in the long term using the Globe Hydrosphere Protocol. Additionally, lablab samples were subjected to qualitative phytochemical testing to determine natural compounds that may be beneficial to the milkfish diet. Overall, the study aims to satisfy the following research questions. First, what is or are the effects of lablab on surrounding water areas, especially milkfish aquacultures and the environment? Second, how does the effect of lablab on the water quality affect living organisms, especially milkfish and aquacultures? And third, what is or are the phytochemical constituents of lablab that may affect living organisms, particularly milkfish? This study measured two major aspects, namely water quality and phytochemicals. To quantify water quality, the study, of course, used the Globe Hydrosphere Protocol wherein water temperature, transparency, pH, electrical conductivity, and dissolved oxygen procedures were used by testing the water using calibrated and wireless Pasco probes. For this, samples were obtained in triplicates from 2 aquaculture sites with lablab and 2 aquaculture sites without. And next, for the crude extraction of lablab, phytochemical testing for the presence of alkaloids, flavonoids, tannins, saponins, terpenoids, and phenols was conducted, and the sample was prepared by washing with water, sun drying, and grinding to a powder. For this study, statistical analysis, a one-way analysis of variance, or ANOVA, was performed to determine the presence of significantly different means in at least 2 sampling sites. For this study's water quality assessments, 4 sites were tested. Sites 1 and 2 were sites with no lablab, and sites 3 and 4 were sites with lablab. For site 1, a pH level of 4.68 was measured with a conductivity level of 20,220.3 microsiemens per centimeter. For its dissolved oxygen level, it measured 5.71 milligrams per liter. And for its temperature, it measured 29.3 degrees Celsius. For site 2, it measured the pH level of 4.70, a conductivity level of 20,200.8 microsiemens per centimeter, a dissolved oxygen level of 4.44 milligrams per liter, and a temperature of 29.1 degrees Celsius. For site 3, a pH level of 4.66 was measured with a conductivity level of 20,136.5 microsiemens per centimeter, a dissolved oxygen level of 7.62 milligrams per liter, and temperature of 30 degrees Celsius. For site 4, a pH level of 4.66 was measured with a conductivity level of 20,185.5 microsiemens per centimeter, a dissolved oxygen level of 11.04 milligrams per liter, and a temperature of 32.3 degrees Celsius. Moving on, the pH levels of the 4 sites recorded similar levels with no statistically significant difference, with the p-value being greater than 0.05. Moreover, for the conductivity, sites 1 and 3 recorded the most statistically different values with a p-value of 0.024. And from all of the sites' conductivity results, it was found that the average electrical conductivity was recorded to be lower in sites with lablab compared to sites without. This may entail the potential effect of lablab in regulating water conductivity. Next, for dissolved oxygen levels, it was observed that there is an increase in DO levels when there is more lablab present. Moreover, sites 1 and 4 and sites 2 and 4 reported the highest statistically different results with a p-value of 0. For temperature, all sites recorded average temperatures within the Southeast Asian Fisheries Development Center, or SAFEDEC AQD standards, which are ideal for milkfish growth. For the phytochemical analysis, alkaloids, flavonoids, saponins, and terpenoids were detected, while phenols and tannins were not. All of these detected phytochemicals have good effects on milkfish growth and quality, such as their enlargement and weight gain. To conclude, lablab has the potential to become an environmentally sustainable alternative to traditional fish feed if its growth is properly monitored and regulated. Its use will be able to reduce nitrogenous waste and prevent further degradation in water bodies and their surrounding areas. However, factors such as weather, climate, temperature, and time of day must be taken into account to be able to further ascertain how much lablab should be grown in an area to prevent algal bloom. Thus, the researchers recommend that more studies should be conducted regarding the effects of alkaloids, flavonoids, saponins, and terpenoids on milkfish populations. That is all for my presentation on my submission to the Globe IVSS 2023 entitled Impacts of Flood Lab or Algal Mat on Water Quality of Chanas Aquacultures in Dagupan City, Pangasinan. Thank you very much for listening, and if you'd like to access our paper and references, please feel free to visit the link or QR code flash. Thank you once again.
Thank you very much for such wonderful presentation. And I know this is really a lot of hard work which has been done, and your presentation excellent, and then congratulations and thank you. Now we can take presentation from Nepal, and then
Actually, Batasha National High School has sent their presentation, so I'm going to share theirs, and then after that we will take Nepal and India. Let me just share. Gabriel, you're ready, right? I'm sharing your presentation.
Yes, but I'm ready.
Okay.
And so, again, good day to everyone. My name is Gabriel Unasin from Asan Hills National High School, and I will be discussing with you all the inhabitants of wetlands and their preservation. Okay, so we already know the use of wetlands and what they can do. They're a vital part of the hydrological cycle, very productive for rich diversity, and they can also do some really amazing stuff like erosion control. They can help effects of climate change and also floods. They can predict floods. So, uh, of course, wetlands are not without threats. Wetlands are being threatened by reclamation and degradation through drainage and landfill pollution. Because wetlands have a stereotype of being wastelands, they are sometimes being drained and repurposed into other types of, usually of arable or agricultural land. So, why are wetlands and its inhabitants important? So wetlands are a crucial part of our environment. Though, as said, they are seen as wastelands, they are not areas that are needed to be drained, filled in, for whatever reason humanity may have for it. And as for the creatures of wetlands, each one of them brings their own unique characteristics, appearances, and their own abilities into the table when it comes to their biodiversity. So here are some examples of some endangered animals that live in the wetlands. First off, we have the water buffalo, or as known in the Philippines as the carabao. So they are standing from 5 to 6.2 feet tall at the shoulder, and male buffaloes actually have a very big horn, as you can see from the picture here, and they have been domesticated for variety of reasons. Usually it is for farming, it is for transporting people and crops. They have also been overhunted. They have been used for their butter fat, their hides, their meat, and also been hunted for their horns. Due to that, they are becoming vulnerable to extinction, and also they're being bred with their domesticated counterpart. Next up is the Chinese egret. So the Chinese egret actually has two types of classifications. A breed adult actually has has a long frilly plume on the backside of their head, neck, and their back. Meanwhile, non-breeding adults lack plumes and has dull greenish legs and bill. So these animals actually migrate to the Philippines during winter. And spend the winter in Philippines. They are mostly being protected by the Las Piñas critical habitat and ecotourism area. However, they have become vulnerable due to the fact that it's difficult to tell breeding adults and non-breeding adults from each other. So moving on, we have the very honorable Philippine duck. So it is a large brown duck with distinctive pale orange face, dark brown, and a dark eyeline. It is wide lowland freshwater and brackish wetland habitats. So this duck is actually vulnerable because it is undergoing rapid continuous decline owing to the extensive overhunting. Because in the Philippines, we have this dish called balut. Balut actually comes from boiled duck eggs, and that is also a reason why we are hunting ducks. So moving on po, the hawksbill. The hawksbill is being protected by the Negros Occidental Coastal Wetlands. And the hawksbill are actually named for their narrow pointed beak and also have a distinctive pattern overlapping the scales on their shells that form a serrated look on the edge. So why are they important? Hawksbills help maintain the health of coral reefs as they remove prey such as sponges from the reef surface. They provide better access for reef fish to feed, and they also have cultural and tourism value because many tourists actually go to different places just so they could see turtles, much like the hawksbill. So of course, I have shown you some animals that are endangered or vulnerable. And there are also some plants that are currently vulnerable or endangered in wetlands. The first one being the green fern. The green fern is a cut-leaf great fern that is native leathery evergreen fern in Ophioglossacea. So this actually has a local name for us, and it's called makahiya. I actually enjoyed this when I was a child. It proliferates due to spores, sends its spores into various areas, and that's how it grows on you. When you touch this plant, it will actually curl up, and that's what makes it very attractive to children in rural areas. The next one is the bog rose. Bog rose is a plant named for its fountain nymph. It's named for the fountain nymph Carytusa, and the flower suggests an animal's open mouth. So this shape of the flower is actually when bees or any insects go onto the plant, it will get a huge puff or many pollen will attach to that insect, and it will cause the plant to be planted anew. However, due to the Philippines having a declining population for those insects like bees, these bog roses are also becoming vulnerable or endangered. The next one is the long-stalk holly. The long-stalk holly is a very hardy evergreen with red berries, pleasantly scented flowers, and showy foliage. We all know holly trees and their red fruits. These fruits are actually relished by birds, and they also really like moist areas. And one reason they could be endangered or vulnerable is because of a thing or a disease called holly tree blight, which causes the holly tree to become very weak. So the last one is the bog asphodel. And the bog asphodel is a sulfur yellow starlight flower of bog asphodel brighten up the brown green peat bogs, damp heats, and moors in early summer. So as you can see, they are usually in the bogs of the wetlands. So enough doomsaying. Here are some of the things or some of the legislations that the Philippine government has taken part of or they have enacted in order to protect the wetlands. The first is the Ramsar Convention. So the Ramsar Convention is an intergovernmental treaty which main mission is the conservation and wise use of all wetlands. Currently, there are 160 nations that have joined this convention as of October 2010, and they have more than 1,900 wetlands around the world, covering over 186 million hectares. And showing you all of these are the 8 Ramsar sites of the Philippines: the Agus Wildlife Sanctuary, Las Piñas Critical Habitat and Ecotourism Area, the Nangwan Lake National Park, the Negros Occidental Coastal Wetlands Conservation Area, the Olanco Island Wildlife Sanctuary, Puerto Princesa Subterranean River National Park, the Sasmuan Pampanga Coastal Wetlands, and the Tubatuha Reefs National Park. Other than that, these are some policy legislations that are relevant to the Philippine wetlands. First is RA 7586, the National Integrated Protected Area System Act of 1992. This is an act providing for the establishment and management of National Integrated Protected Area System. So basically, this act will actually protect a certain area and will be owned by the government so that it can be protected, preserved, and conserved. The next is RA 9147, the Wildlife Resources Conservation and Protection Act. So this act is actually very general. It applies to all animals. However, it is still relevant in wetlands as it aims to conserve their home and also protect the wildlife of wetlands. Next is Executive Order 533, adopted integrated coastal management as a national strategy to ensure sustainable development of a country's coastal and marine, environmental, and resources, establishing support mechanisms for its implementation. This was added in 2016. So those are some of the legislations that the government has done. Now, how do we as individuals protect the coastal wetlands? This is in reference to the U.S. Environmental Protection Agency, updated as of January 16, 2024. The first is that we should participate in programs that help protect and restore wetlands. You can also contact local, state, or federal agencies, community groups, and other organizations for their programs. Number 2, report illegal actions such as unauthorized landfill or dredging activities to the government authorities. Number 3, pick up litter and dispose in an appropriate trash container. Keep the surface areas that storms drains clean from waste, toxic chemicals, fertilizers, and motor oil, which can eventually reach and impair our wetlands. Number 4, use native species for planting trees, shrubs, and flowers to preserve the ecological balance of local wetlands. Number 5, using living shoreline technique that makes use of plant roots to stabilize soil if your own waterfront property and shoreline or riverbank needs to be stabilized. 6, avoid wetlands if you are expanding your house or installing a shed. So number 6 is very basic. Just avoid wetlands if you are living near one. 7, use phosphate-free laundry and dishwasher detergents. This is because phosphate encourages algae to grow and it can actually suffocate aquatic life. So number 8, use non-toxic products for household cleaning and garden care. Never spray lawn and garden chemicals on a windy day or on a rainy day to wash the chemicals away into waterways, which will lead to the wetlands. 9, use non-toxic products for household cleaning and lawn and garden care. Never spray lawn and garden chemicals outside on a windy day. So, same thing. So 10, enjoy the scenic and recreational opportunities coastal wetlands offer while preserving their integrity for future generations and minimizing the use of heavy equipment and staying in designated visitor areas where available. There's an old saying when it comes to traveling in parks that I've heard. It is take only pictures, leave only footprints. And I think that is a very good saying to follow when it comes to visiting scenic recreational areas such as coastal wetlands. So here are some recommended organizations for youth and other students alike that are focusing on wetlands preservation. The first is the Society for the Wetlands Conversion of Philippine, the Society for Conversion of Philippine Wetlands, and their website can be found here. The second is the Wetlands Education for Students and Teachers, and their website can also be found here. So thank you for listening to my presentation. I would like to thank the help of these people in making my presentation possible. Thank you all.
Thank you for this very elaborate and very interesting, informative presentation on the wetlands and the activities which you have in Philippines. Thank you very much, and congratulations for such a wonderful presentation. Now we have the last presentation from Nepal.
No, the second last, because BCM Arya is also here. So Nepal is the second last presentation. It's Amity Public School.
Now, you want to say anything before that?
Who?
From Nepal. Yes. Yogender, hello.
Let me see. I'm not seeing.
Please, if you can take BCM first, because our send-off time of students are about to be.
Actually, the Nepal is waiting for a very long time, so we would like to take Nepal.
10:30. Nepal.
Yep.
Yogender is not here?
He is, yes.
Yogender? Okay. Okay, then make the presentation.
Am I audible?
Yes, you are.
Very warm and cheerful good morning, good afternoon, everyone. We welcome you all to our presentation about wetlands. First of all, I'm very thankful to ECA Nepal and Globe Program for providing such an ample opportunity to share our students and wetlands. Hi, I'm Jay Shetrai.
Hi, I'm Sojalaga.
Hi, I'm Prashant Patel. Hi, I'm Vijay Vijay.
Hello, I'm Shravan Manwar.
We are the members of Amity Nature Club and the Duke of Edinburgh International Award, Amity Public School, Kakarda, Japan, Nepal. So what actually are wetlands? Wetlands are the areas where water covers the soil or is present either at or near the surface of the soil all around the year or for varying periods of time during the year.
Objectives of our project: to analyze difference between pH in wetland, EC difference in wetland, temperature difference in wetlands. There are many wetlands in Nepal, but we analyze the nearest wetland from our school. Jamunbari wetland, 36 km to the west from our school. Kalika wetland, 7 km to the west. Ledap wetland, 10 km to the north. Here we can see the pictures of wetland.
So we have collected some of the pictures of birds around wetlands. So let's have a look at them.
Let's see the statistic of pH, EC, and temperature of the wetland. On 6 September 2022, we went to the Ledap. Here were 3 lakes. In first lake, the pH value was 7.7. whereas EC was 0.02 and the temperature was 29.9 degrees Celsius. In second lake, the pH value was 7.2 and EC was 0.03 and the temperature was 29.9 degrees Celsius. In the third lake, the pH value was 7.2 and the electroconductivity EC was 0.03 and the temperature was 29.9 degrees centigrade. On 7th September 2022, we went to Kalika wetland. pH value of the wetland was 7 and the EC was 0.02 and the temperature was 38.12 degrees centigrade. On 11th September 2022, we went to Jamunbadi wetland. The pH value of the wetland was 7.4 and EC was 0.02, whereas temperature was 32.7 degrees centigrade. On 17th December 2022, we again went to Kalika wetland. The pH was 8.5 and the EC was 0.07 and the temperature was 20.1 degrees centigrade. On 24th December 2022, we went to Jamunbadi wetland. The pH value was 8 of the wetland and the EC was 0.02, whereas temperature was 27 degrees centigrade. On 31st January 2023, we again went to Jedhav wetland. There were also 3 lakes. In the first lake, the pH was 7.9, EC was 0.03, and temperature was 22.2 degrees centigrade. In second lake, the pH was 8 and EC was 0.03, and the temperature was 22 degrees centigrade. In the third lake, the pH was 7.8, EC was 0.03, and the temperature was 22.2 degrees centigrade. On 1st January 2024, we again went to Jedhav wetland. In the first lake, the pH was 8.7, EC was 0.03, and the temperature was 19.1 degrees centigrade. In the second lake, the pH was 8.3, the EC was 0.03, whereas the temperature was 19.9 degrees centigrade. And in the third lake, the pH was 8.2 and the EC was 0.07 and the temperature was 20.7 degrees centigrade. In 1st January 2024, we also went to Kalika wetland. The pH of the wetland was 8.7 and the EC was 0.02, whereas the temperature was 29.9 degrees. On 2nd January 2024, we went to Jamunbadi wetland. The pH value of the wetland was 8.4 and the EC was 0.02 and the temperature was 20.3 degrees centigrade.
Let's have a look on analysis between EC, pH, and temperature. Month September 6, 2022 and 31st January 2023. Hiletap wetland. The pH value, pH difference of first lake of Hiletap wetland is 0.2, EC 0.01, and temperature difference is 7.7 degrees Celsius. Second lake, the pH difference is 0.8. There is no difference in EC and temperature difference is 6.0 degrees Celsius. Third lake, pH difference 0.6. There is no difference in EC and the temperature difference is 7.7 degrees Celsius. On 7th September 2022 and 17 December 2022, Kalika Lake wetland. pH difference 1.5, EC difference 0.05, temperature difference 18.11 degrees centigrade. 11 September 2022 and 24 December 2022, Jamunbadi wetland. pH difference 0.6. There is no difference in EC and temperature difference is 5.7 degrees centigrade. 17 December 2023 and 1st January 2024, Kalika Lake. pH difference 0.2, EC difference 0.5, temperature difference 0.1 degrees centigrade. 24 December 2022 and January 1st, 2024, Jamunbadi wetland. pH difference 0.4. There is no difference in EC and temperature difference is 6.7 degrees centigrade. 31st January 2023 and 1st January 2024, Hiletap. First lake, pH difference 0.8. There is no difference in EC. Temperature difference 2.1 degrees centigrade. Second lake, pH difference 0.3. There is no difference in EC. Temperature difference 3.1 degrees centigrade. Third lake, pH difference 2.2. EC difference 0.5. Temperature difference 1.5 degrees centigrade.
Here are some pictures that were collected during the data collection. Lastly, we are very thankful to the GLOW program for providing us the opportunity and the technical support for implementation of the GLOW program in Amity Public School, Japan. It has brought immense difference in our studies and knowledge beyond the classroom and doing science. It has changed the identity of the students and the school in community as well. Thank you very much.
Okay. Thank you very much, guys, for your presentation with a lot of information and data and the field work which you have done. Great work you have done. Thank you very much. And I know you got the information about this symposium very late, but still I appreciate your concern. And then thank you very much for this information. And I am sure that the lake which you have studied, you will continue. I see Yoginder is here. Yoginder, you want to say something or no? Yoginder, I saw you are here. Yoginder, no comment, no suggestion?
Yeah, I'm here, Bangladesh.
Yes. Okay.
See, we got information much more later, and we were very busy with our activities over here. The students had just completed their holiday, and
Yeah.
it was quite
Yeah. And
within
also
2
very
days.
cold. And also very cold also. Yeah. Okay.
Yeah.
Okay, now we do the last presentation from BCM.
Thank you very
Yeah,
much
the
for
last presentation is from BCM Arya International School from Punjab, India, and their topic is, I think they'll announce their topic. I'm sorry if it's not.
Good afternoon, all. We are from BCM Arya Model Senior Secondary School, India, and our students are ready for the presentation on Harike Wetland.
Good afternoon, everyone. I am Ibne.
I'm Talin.
And we are from BCM Arya Model Senior Secondary School in India. So today we're going to present about Harike Wetlands. So for the introduction, Harike Wetland is also known as Harike Patan, and it is one of the largest wetland in northern India. It is situated at the confluence of rivers Beas and Satluj at Firozpur Amritsar border. It is a paradise of every bird watcher and is situated in the heart of Punjab, which is a state in India. It was recognized as a Ramsar site in 1990.
Now I'm going to tell you about its vegetation. Harike wetland is the home to rich vegetation and comprises Icoria crassipus dominates in about 50% of the area. Azolla are sparsely seen in open water areas. Nelumbo nucifera, the lotus, in the prominent rooted floating vegetation. Ipomia aquatica is at the lake periphery in the shallow regions. Delbergia ciso, Acacia nilotica, Ziziphus figus, Prosopis julifera.
In large clumps and other trees are painted along the alignment. Najos hydrilla, Ceratophyllum, Puja Vallisneria, and Charals are the species of submerged vegetation. Typha is the dominant emerging. So now we're going to talk about the rich wildlife. The Harike wetlands are home to a wide variety of flora and fauna and fertile farmlands rich with biodiversity.
Harike records over 360 species of migratory birds and several animals too. Bird species like northern pintail, medieval are found in Harike wetlands.
It is home to various species of animals such as the smooth Indian otter, the jungle cat, Bengal tiger, jackal, Indian wild boar, common mongoose, and many more. So in the end, for the conclusion, we would like to say thank you.
This was all for today. Thank you so much.
Thank you, BC Amaria, for your presentation. Again, before concluding, I would like to thank you all the participants for participating in today's virtual symposium. And we will be sending the participation certificate for teachers and as well as students to their country coordinators positively by tomorrow. And we would very much recommend you to upload your presentation or resubmit it in the journal format so that we will be able to compile everything and send it to Rod for final editing and everything. And then it will be published on the website as well as circulated in the region and globe at large. So we will very much recommend students to go on the Philippines page, and they have a guideline over there for how to write the paper and what is the particular format for submitting the research paper for journal. And I'm also putting a link in the chat box for everybody who want to send their papers. So if this is all you want to do, you can go it from there also. And I would like to request Dr. Desh for the final comments.
Okay, any country coordinator want to make any comments, please? I think some of country coordinators still they are here. And if you want to make any comment, please do that. So that helps us for our planning activities in future. Anybody wants to make any comment? Rod, you want to make any comment?
No, I just wanted to commend everyone. In this.
Okay, this is around 5 hours now. We started 4.5 hours. Now we started our activity today that have been going a long way.
Some of the hard to imagine that these are, but it is so also.
Okay, country coordinators, I request you to send a country report for the regional meeting, which we plan to have in Maldives. We are waiting approval from GIO, and after that, we will be sending you the formal invitation. If you like to have the invitation or something for your ticket and everything, we would like to do that. And also, let us keep in touch. Well, I don't have to say much because country coordinator, Yoginder, you are here, I can see you. Would you like to say something? I cannot hear you, Yoginder.
I think his mic is not working.
Cannot hear you, Mike. Yoginder cannot hear. No.
Is there anything any student or anybody, any participant would like to add?
Can you hear me now?
Yeah,
Oh,
yeah,
yes,
we can
yes, now I can hear. Yes.
Okay. I'm just saying that this one day, Wetland Day is not enough for our presentation. We should celebrate Wetland Week celebration so that every school will get presentation opportunity. So this is wonderful. And when I go through this all presentation, I thought that now Globe students are encouraging and they are motivating on doing Globes. So it's quite encouraging for us also. And just to mention here, today just we decided our children club conference in Nepal from May 20 to 24. So we are planning to do this national conference in Nepal, and that time, if our Globe schools are interested, they can participate in Nepal. So this is invitation for you all.
That is in Kathmandu or somewhere else?
Yeah, it is outskirt of Kathmandu in Dhulikhel.
In Dhulikhel?
In Dhulikhel.
Okay,
Yeah.
that's a That's a tourist resort.
It's wonderful resort, yeah.
Yeah, I know. When it started sometimes 20, 25 years back, I was the first visitor there.
Yeah, that is near to that area, yeah, in Boniba, yeah.
Great. I recommend that is a beautiful place, and then maybe, please try to visit Nepal, and that is a good weather there that time, and also not very far from Kathmandu. One can go by bus, taxi, or something like that. It's a wonderful place.
Yeah, I think that's my statement for today. Thank you very much.
Okay. Okay. And next time we will plan up a wetland week. I think that's a good idea because 5 hours sitting in a long is a very long.
No, more than me, I think it's for the student because I know most of the schools are closed and they are still in school because of this thing. So thank you all for your patience for us today, and thank you for joining this event and making it a great success for us. And we will be uploading this recording in our YouTube channel, and we will be sending the link to you via email or via our social media post. So if you are subscribed to our channel or our social media, you will get to know about this recording sent live on the YouTubes and everything.
I think somebody from Sri Lanka want to say something.
Yeah, of course, the floor is open for anybody who want to make a comment.
Any comments? Garg, you want to say something? Okay. As I think people are feeling shy. Dylan from Pulau, she's also here.
Yeah.
Please make point to come to Maldives.
Oh, yes. Thank you, Dr. Desh. I miss you guys. And yes, of course, definitely. I will find time. If you guys send me an invite, of course. So, yes, definitely.
One person from one country, like country coordinator. Okay. Victoria, I think she has gone. Maybe it's okay, long time. So maybe we can break our meeting here with all the thanks to you, everyone. And also, please, I'm thankful to all of you sending the condolences message for me and my family. We had a great loss anyway. Thanks, everyone. Thank you very much. So we call off the meeting now.